So thanks very much. It's great to see such a full room for the afternoon session. The afternoon session on day 2 of Research Week 2025. We're delighted to have the LDCRC back with us again this year. Obviously, it was such a success last year. They came back for more, which is great to see. So just on the housekeeping front, if you can have your mobile phones on airplane mode or on silent, please, just to give the speakers a fair chance. And also, as you can see, there's plenty of photography and videography taking place today. So if you don't want your image included in any of that, please contact one of the people in the bright pink t-shirts and we will look after you. So I'm going to hand straight over to Professor Aideen Culhane, who's going to chair this afternoon session.
Thanks everyone so much on coming out and being here today, especially when the sun is shining. Really appreciate it. Thank you so much, and hopefully we will listen to some incredible science today, and I think it's going to be worth it. We have some really exciting announcements and things that are happening in the LDCRC that we will discuss later on. I just wanted to mention one brief thing, which is next Thursday afternoon, please sign up your labs. There's a couple of people I'm looking at that I haven't seen their sign up yet. We are doing a really innovative thing which will try to get labs across the university who are working in cell culture and cells, spatial biology, organoids, 3D printing, cell matrix biology, get everybody together so you can see what's happening in physics, what's happening in engineering, what's happening in medicine, what's happening in biology. Everybody's going to talk about their labs really briefly, and then we're going to have a physical walking tour where we actually walk from lab to lab to lab so you can see what's happening in other labs across campus and seeing what expertise they're bringing to this problem, so how we can develop better in vitro culture systems for patient tissue, for cells, so that we can get better functional biology assays and better drug response measurement systems for patients. So really cool. Sign up your lab. Tricia is here. Tell her you're signed up and be part of this really exciting, innovative thing that we're going to do. And then that's going to follow on by a one-day intensive kind of hackathon where we present problems. And we have identified a really fun grant application, which is the UK Grand Challenge grant. Really big. These are like 20-30 million grants. And we're going to put together an application with UL as a partner, which will go forward for the Grand Challenge grant. So I want everybody with their best thinking caps on when we tour the labs and when we have our one-day hackathon. Okay, so this is going to be really exciting. Okay, so that's the exciting thing we're going to do. But I am more even than that excited to introduce our next speaker. Okay, so I have to get the glasses on. So I am honoured to introduce Professor Donald MacDonald. He's a native of Limerick, and he was winner of the Young Scientist Award in 1978. So that's so cool. He earned his degree in biochemistry from the National University of Galway and in 1983, and then he did his PhD in cell biology at Bayer College of Medicine in Houston in Texas. He is the Glaxo Wellcome Distinguished Professor of Molecular Cancer Biology at Duke University, which is in North Carolina in the US. He is an incredible figure who leads international breast cancer research, and he pioneered from start through to FDA and EMA approval of a new breast cancer drug. And this is a drug for selective estrogen receptor degrader. It's called Elastrit, and he's going to tell us about it and how to go from basically the lab to having impact in patients, and that's so exciting. Even better, this new drug, it's taken orally. So for patients, that is massive. His groundbreaking work bridges cutting-edge molecular biology with translational oncology, driving innovation in targeted hormone therapies that are transforming treatment for women with cancer. His career exemplifies the power of scientific discovery, and his keynote will offer insights into the future of cancer treatment. So with further ado,
So that was actually like a eulogy, right? It's funny, just looking at so many students in the audience, PhD students and that, you'll enjoy the story that it'll tell you, which is a very true story. I was telling Nadine and a couple of others, my mum was down in the Park nursing home down here, and I walked in the other day, and she says to me, she says, "Can they find out on that internet thing that you're not a real doctor?" And I says, "Mom, what have you done?" And she says, "Well, Mrs. Moriarty, her son graduated from UL, the medical school, and went over to North Carolina, and she's always talking about him. So I told her that you run the Duke Cancer Center." I says, "Mom, for crying out loud, I'm a very accomplished researcher. I don't run, but are you not proud of me? And she turns around, she says, "Son, we are, yeah. But your father and I were very disappointed when you decided not to become a real doctor." So I hope I don't disappoint you guys. So today what I want to do is, actually Aideen kindly gave the punchline, which is great, is a 30-year search for new drugs for metastatic breast cancer. I could give the same talk, luckily, for a 30-year search for new drugs for metastatic prostate cancer, but I decided to talk about the drug that is already approved for use in the United States and just last September for use in Europe that targets the estrogen receptor in late-stage disease. The reason I chose this story to tell today is because exactly what Aideen said. It started off as a nubbin of an idea when I was doing my postdoctoral fellowship back in 1988. That's a long time ago. And then basically then we went through many ups and downs. I'll talk to you about a few of those. And then success. And success looks like drugs in a bottle that are brought into the oncologist's office. So I don't know what's required in Ireland, but I'm absolutely required by my university to declare my conflicts of interest. And obviously, I have a conflict of interest in the royalty stream from elacestrant and Sermonex, which is lasofoxamine, a second drug that we have in the clinic. So I want to bring everyone here together to the same page with not a too complicated a slide, but it's really telling the audience really that there are three types of breast cancer. And they're defined not only in terms of their biology, but in terms of the way we treat those cancers. The majority of cancers, those of which mostly are cancers of women over 50 years of age, are what's called estrogen receptor positive cancers. And you've heard all this talk about precision medicine and the advent of precision medicine. I would put to you that ever since we've been using endocrine therapies, we've been using precision medicine because what we're doing is we're treating estrogen receptor positive breast cancers. I'll talk about the evolution of these therapies for that disease as we go through this talk. But I'll just briefly mention the other two cancers because there's a big success story and a big challenge in the other two. HER2 positive. HER2 is a signaling receptor that's expressed on about 15 to 20% of cancers. That is interesting because Herceptin and pertuzumab are two drugs that came out of Genentech and AstraZeneca that actually target HER2. And it's interesting because in that disease, we now use the word cure, not just treat. And that has been really a remarkable success. And then finally, the challenge is triple-negative breast cancers, defined as these are cancers that are particularly aggressive, but they're more common in young women, and they're also more common in African-American populations. Unfortunately, as of today, we do not have any targeted therapies for triple-negative breast cancer, although the recent advent of immunotherapy has shown some promise. But as I promised in the beginning, I'm going to talk about estrogen receptor positive breast cancer. And I do like to talk, to actually use a few slides to talk about the background and to get you all interested in this receptor as a therapeutic target. So for those of you who have forgotten your biochemistry and Stryer, this is a diagram of how the estrogen receptor is thought to work within cells. The estrogen receptor is what we call a ligand-regulated transcription factor. In the absence of hormone, it sits in the cytoplasm associated with heat shock proteins. Upon binding a ligand, it undergoes a conformational change. That was something one of my students showed 25 years ago. It forms a dimer, enters the nucleus, binds to target genes, and turns their transcription on. Now, we're not going to go through the details of transcription today, but I put this up here to describe where the two primary interventions that are used to treat breast cancer work. You have a series of drugs which are called aromatase inhibitors. These are the frontline therapies. They block the last step in the conversion of testosterone to estrogen in women. And estrogen, of course, estrogens are the primary drivers of the activity of this receptor. There are frontline treatments now, pretty much worldwide, for the treatment of breast cancer. However, the second one, and the one that kind of really wholly started this whole field, was a drug called tamoxifen. And tamoxifen binds to the receptor at the estrogen binding site and competitively blocks the binding of estrogen, thereby shutting off the receptor. What most people don't realize, and I think it's a very interesting story, is that tamoxifen was never developed for breast cancer. It's a failed oral contraceptive. It was developed at ICI Laboratories, AstraZeneca now, and it was discovered, as I said, by Mike Harper as a drug that actually would work to shut off estrogen signaling in the pituitary hypothalamic axis. Sorry, it's there. Pituitary hypothalamic axis. And in doing so, they thought it would be a contraceptive. Well, actually, it got the nickname the whoops drug because it caused hyperovulation, and women ended up getting multiple Paris births. But Craig Jordan, okay, who was a postdoc, who will tell you himself, he has passed away recently, so I'm not going to tell anybody anything. But Craig just passed away, but he was just a remarkable scientist. Craig actually, he will tell you himself that he was a failing postdoc. Okay, and his mentor shipped him over to ICI to learn something about estrogen receptor pharmacology. And he basically said, he discovered that this molecule, tamoxifen, was an estrogen receptor antagonist. And he ended up getting this drug popularized, socialized enough within the company to get into clinical trials in the adjuvant setting for breast cancer. The results were absolutely remarkable. Okay, this is just this slide is already about 15 years old. There hasn't been an update for a while because tamoxifen is not used really much in frontline anymore. But all I want you to say is that in the left-hand side of this curve is early stage breast cancers, and this is later stage breast cancers. And what you can see by just looking at these lines, that tamoxifen, the addition of tamoxifen, improved disease-free survival, progression-free survival, by about 50% in both cases. And then most importantly, the drug was only delivered for 5 years and the effect endured. So it's interesting, Craig was actually given the Brinker Award at the San Antonio Breast Cancer meeting. This is about 15 years ago, and I was at the lecture. And Sir Richard Peto introduced him by saying that this man, Craig Jordan, was responsible for saving 800 million years of women's lives. Now that's a legacy. Not bad for a guy who considered his postdoc was failing. But the thing reason I mentioned that this was not a breast cancer drug initially was to say that what happened was we all went back then and said, well, gosh, tamoxifen is not that good an inhibitor. It doesn't bind with very high affinity. It also has what's called partial agonist activity. It mimics estrogen action in the uterus, and that can actually increase the risk of endometrial cancer or uterine cancer. And so when I started this field, the challenge was to come and build a better mousetrap, a better tamoxifen. Well, I was personally involved in droloxifene, idoxifene, and pipendoxifene. Lilly developed an amazing drug, arzoxifene. The bottom line is none of them worked. None of them worked better than tamoxifen in the front line, and none of them worked post-tamoxifen when tamoxifen failed. And the title really describes where I'm going to go here. And that is that the simple model of estrogen action I told you about is that the estrogen receptor drives the transcription of genes that are responsible for cell growth, proliferation, and tumor metastasis. And the model also basically suggested that the receptor existed in an on or an off form. The on form drove that. The off form, which was the tamoxifen-bound form, didn't drive that. And so it didn't make any sense to us that if we made higher affinity compounds that didn't have any agonist activity, why were they not working better? So there was a clue, and I took out the slide just to be short. There was a clue from a paper that was published in The Lancet. It was a case report where the authors had described a process called tamoxifen flare. Okay, and this was really a paradox. You give this compound, which is an anti-estrogen, and you give it to patients, and the tumors actually grow faster than if you didn't. You take away the drug, and the tumors basically regress. So wait, this is an antagonist. So why would an antagonist actually stimulate the growth of the tumors? So this brings me to the first student story, and this is a student, Caroline Connor, who came into my lab, and I gave her that paper, and I said, listen, I don't know what we're going to do, but we're going to build a project around this case report and try and see if we could, come up with some way to model it. And so this is, every presentation, every presenter has their favorite slides. And this is one of mine because it was really an aha moment and it helped us to drive our program further. So this is a very simple experiment. You take a cellular model for estrogen receptor positive breast cancers. Those of you who work in breast cancer will know it. It's the workhorse MCF7. And you put it into mice that are supplemented with estradiol, and the tumors grow, shown in blue, grow very, very well. You now come and you add with tamoxifen, the anti-estrogen, on top of that, and you suppress the growth of the tumors. No big surprises here. But what Caroline did then was, and again, no big surprise, she let this red bar, these are the inhibited tumors, she let those go to the point of resistance. We knew it was going to happen, okay? And resistance occurred. What the bright thing she did, which I actually didn't, to be honest with you, didn't think about doing, was to take the resistant tumors and reimplant them back into mice and now look at their growth as a function of tamoxifen. This was the aha moment, because it turns out that the tumors, not only were they resistant to tamoxifen, predictable, they actually depended upon tamoxifen. So now if you took away the tamoxifen, they regressed. So this basically suggested that the simple on-off model really couldn't be corrected, where it's not correct. Because under some circumstance, the circumstance we have created here in the animal model, we have now found a situation where tamoxifen can actually function as an agonist and not an antagonist. So there was a switch in its pharmacology, all right? And its pharmacology was not necessarily that of an agonist. In fact, we don't use the word, an antagonist. We don't use the word antagonist in our field anymore because everything is context specific. Every molecule that's an antagonist can function as an agonist in some regard, and every molecule that we define as an agonist can function as an antagonist in some regard. So we basically just call them selective estrogen receptor modulators. So I'm going to summarize 20 more years' work in one slide because we actually figured out what was going on. And this is I think, one of the things, I can actually boast all I want because I didn't do any of this physical work myself. My students did. But this is where the concept of functional allostery developed. And I'll explain to you what that is in a second. So remember where I started off this. The receptor could exist in an on or an off form within the cell. But that didn't explain the in vivo pharmacology of these drugs. So what John Norris, who was another student in my lab, discovered was that the receptor doesn't just exist in an on or an off form, that the receptor is actually very malleable, okay? And depending upon the subtle differences in the structure of the ligands that binds, tamoxifen is different from raloxifene, is different from orzoxafen, the shape of the receptor changes, okay? So I always remember we submitted a paper, I'm not going to tell you how we did it, but it was a really cool technology. It was published in Science in 1999. I didn't tell you how long ago this is. But I always remember reviewer number 3. And reviewer number 3 came back and said, the McDonald Laboratory has discovered a technology to identify microallosteric changes in the estrogen receptor structure of unlikely importance. But actually, we went and showed that that was important because what different shape changes in the receptor did was allowed the receptor to engage different cofactors within the cell, okay, and different transcription factors. And then the third part of that thing was model was to show that different receptor cofactor complexes could give you different biological activities. And so a whole new world of estrogen receptor pharmacology was opened up to us at that particular point in time. And we came up with 3 rules that guides the pharmacology pretty much of every ligand. This is now held to progesterone receptor, the androgen receptor, the glucocorticoid receptor. It's the same model. Shape matters. The shape, the subtle differences in the shape of the ligand alters the receptor shape, allowing different interactions with cofactors, and the absolute, relative and absolute levels and activity of these cofactors difference between cells. And so then this allowed us to try and come up with a model that explained Caroline's mouse data. And the model was very simple. And that is that initially, that what tamoxifen did was drove the receptor into a conformation or a shape that it couldn't find a cofactor in the cell that it needs to drive transcription, so the tumor was inhibited. But because of the huge selective pressure that was on the cell, okay, there were probably a subpopulation of cells within that tumor that had a cofactor that said, I don't care, but that shape is fine with me, and I'll interact with it, and away I go, and I drive the tumor. And so the hypothesis then was, well, gosh, if it was as simple as that, we should be able to screen, for compounds that would drive the receptor into yet a different shape, and we start the whole process over again. Well, I wouldn't be building myself up like that if this is not the way it worked out. So at that time, industry-academic collaborations were a lot easier. So this would have been about 1999, were a lot easier. And we actually got access to the Wellcome collection of estrogen receptor modulators. They had a program, nothing to do with cancer, but they had thousands and thousands of estrogen receptor modulators, and we got them. And I'm not going to go through all the data. Anybody wants it, they can read the papers on this. But we screened looking for compounds that would drive the estrogen receptor into a different conformation. And the first drug we found was a drug called GW, Glaxo-Wellcome 5638. So this was discovered in my lab in the year 2000. So we could not get them interested in this drug for love nor money. And so we did something which was very unusual, and we licensed the drug. We being Duke. We licensed the drug from a pharmaceutical company. And we brought it into Duke and played with it for about 2 years. We didn't change the structure. We worked on the formulation, we worked on the biology, we worked on the pharmacology, and then I'll tell you what we did with it afterwards. But let's start here on the right-hand side first. So this is the model I showed you where tamoxifen stimulates the growth of the tumor, shown in blue here. And if you add now this GW compound, it actually suppresses the growth of these tumors. So remember what we've done. We've identified a compound that drives the receptor into a shape that disengages the known offending cofactors, and we started off this process of sensitization all over again. So the other thing is that we now, and I didn't show you the data, we solved the crystal structure, okay, of this drug bound to the receptor, and we actually showed that it did actually drive the receptor into a different shape. And one of my other bright graduate students, Ashini Wajaratne, I always joke she has the biggest name in science, definitely the most syllables, letters. But anyway, Ashini walked in and she was so excited when she saw the crystal structure. And she said, Donald, have you seen it? I said, yeah, I saw it. It was interesting. She said, all the changes, I said, the changes were very subtle. I said, disappointingly so. She said, no, they're fantastic. Now, this is the point where I'm getting embarrassed because I'm the one who trained as a biochemist, okay? And she said, there are 3 leucines down here that are twisted from the hydrophobic to the aqueous environment. And I'm still staring at her, waiting for a punchline here. And she said, that means that the cell is going to recognize this as a denatured protein. And I said, oh my God. So she went and she actually did what's called a Western immunoblot blood analysis. And all you need to show here is that in these cells, when you add this compound, not only does it competitively bind to the receptor and block estrogen binding, but it denatures the receptor. The cell sees it as a damaged protein and gets rid of it. The field of selective estrogen receptor degraders or downregulated was born. And that was a graduate student, Ashini Wajaratne. This paper, I mean, it's funny. It was she wrote up the paper, and now this is where I get good, is trying to take her excitement and making it even more exciting. And this paper basically then is referenced as kind of where this whole field started from. So now we'd basically found a competitive antagonist, okay, that also degraded the receptor. Well, it turns out that was very timely because at that time there were lots of pieces of data emerging, especially in the cancer field, that the receptor does not always require estradiol to be activated. Now, that was a bit heretical at the time. I remember it was my mentor who discovered it, Bert O'Malley, and I remember him standing up at the Endocrine Society meeting and basically saying this. And I remember talking to my friends thinking, I think Bert's lost it, okay? But it turns out that actually he was right. And he and Ken Korak and Benita Katzenellenbogen and all these labs, Jeff Green's lab, all discovered that the activation of these pathways, extrinsic or upstream of the receptor, can lead to phosphorylation of the receptor, allowing it to be activated absent a canonical ligand. And so what companies started doing was combining endocrine therapies with a MAP kinase inhibitor or an AKT inhibitor and things like that. And all they ended up doing was playing whack-a-mole because you inhibit one and the other one comes up. And so the idea then became, let's just degrade the receptor. So what happened to this wonderful drug, a Taxol, that we developed? So we out-licensed the drug to DuPont Pharmaceuticals, and with Patrick Nevin in Leuven in Belgium, we put it into clinical trials, and it worked fantastic. And then the worst possible thing can happen. DuPont got sold to BMS. And BMS gave the drug what's called an NPV, a net present value of 0. They basically said the world does not need another endocrine therapy. They killed the program, and a drug that actually had worked in a phase 1/2A clinical trials was killed. Let me just tell you, I'm going to tell you the success story in a second, but the successive drug was no better than the drug that they killed. So it turns out that there were some more enlightened people than those in BMS. And when we published the structure of our compound, 11 different companies basically went after that, took that structure and basically did what's called a fast follower program, i.e., how can we get around their patents? So Genentech, Sanofi, Bristol never went back into it. They couldn't, they were too embarrassed. Glaxo, all of these companies went back into it. And I'm going to tell you in a few minutes where the state of the field is. However, I really wanted today to keep highlighting basic research and bright young people, okay? Because we continued, even I can't even tell you how devastated we were when we heard that this drug was killed, because we knew in our hearts of hearts that was, we thought anyway, that was the best we could do. So Suzanne Wardell, who was a postdoctoral fellow at my time, she's now an assistant professor in my group. I couldn't remember if her promotion went through or not yet, sorry. She's assistant professor in my group. She was reading the literature and saw a very interesting paper. And this paper was describing a drug called RAD1901. So this was an estrogen receptor agonist, at least that's what it was called. It describes an estrogen receptor agonist. And it was being treating, it had failed in clinical trials for dementia by a Japanese company called Eisai. So then Radius Health licensed it and said, let's test it for hot flashes. And it gave a really, really unusual dose response. So this is the number and the severity of hot flashes on this axis. This is time. What they found was black is the placebo. Anybody who's on hot flash trials know the placebo effect is about 40%. And if you come along and you give 10 milligrams per day of this drug, you suppress the hot flashes. And if you give higher concentrations, you actually don't have any effect, or in fact, in some cases, you might actually increase the hot flashes. So they kill the drug, dead. Suzanne saw this, and we're pharmacologists. We love weird pharmacology because there's always some biology there's always some interesting biology underlying weird pharmacology. And so Suzanne, anyway, to make a very long story short, we got the drug. We made it ourselves, I think. No, we didn't. We actually got it from the company. And we went and started doing some profiling. Within 2 weeks, we had a drug that we found degraded the estrogen receptor. So here is fulvestrant. Fulvestrant is an injectable SERD. It's a horrible, horrible drug. Women have to take 5 ml injections into the buttocks every month. It's and ulceration and pain. Pain with the injection and ulceration thereafter is a big issue. So, but it is the standard of care. Was until our drug was discovered, the standard of care. But anyway, to make a long story short, you see fulvestrant degrades the receptor. This is a Western looking at the degradation of estrogen receptor in the tumors by Western, and this drug also degraded it. So let me just reiterate, this drug is a competitive competitive binder. I'm telling you now it's an antagonist, even though it was originally developed as an agonist. Remember I said an agonist is not an agonist, an antagonist is not an antagonist. And now it also drives the receptor into a conformational state that causes its degradation. So if you actually come and look at the tumors themselves and you quantify the receptor in individual tumors now, this is just looking at them in bulk, you can see that very, very dramatic dose response curve with respect to downregulating the tumors. If you look in the mouse uterus, which is just to see is this species related or not, and the reason anybody interested in drug discovery, you want your drug to work in mice as well, because otherwise the preclinical evaluation is really, really difficult if it doesn't. And so it works, it degrades the receptor there. Do you remember that model where we stimulated with tamoxifen? It also works there. So we went and filed patents on the repurposing of this drug for metastatic breast cancer. Its patents were just about out for the structure. And we got them. We got them both in America and in Europe. And then the company got interested. But by that time, they'd already sued us. We'd won the lawsuit because they said that we stole their private information. That wasn't true. We were the ones who generated the information. But the bottom line was the trial, the study went forward, and it was called the Emerald trial. And the Emerald trial basically showed that this would work in patients with advanced metastatic estrogen receptor positive breast cancer who had failed standard of care aromatase inhibitors. And this is kind of the boast of the slide. This is what we do it for. Okay, I forgot to mention poor Eric in this one, but Eric Nelson was a postdoc in the lab who had helped Suzanne in this study. And the drug was approved in Europe last September, and it was approved in the United States last January. And so we have heard that so far it's actually, it is the fastest selling. Now selling, I'm not necessarily focused on the money, but money tells you many times the prescriptions have been read. It is the fastest selling breast cancer drug in ER positive in 20 years. And so we're very, very, very proud of this. So the drug is called elacestrant. Okay, sorry. Or its trade name is Orserdu, if you ever want to look it up. But anyway, that is. I'm not sure I thought that we could do any more. But I had another graduate student. So I had a graduate student who I think every student in this audience will empathize with. Her primary project was failing and failing fast. Okay, she's in year 3 of her project. She's got a 5-year fellowship. She's, she doesn't know that light at the. She knows now that the light at the end of the tunnel is an oncoming train. Okay, it's not the end of the road. And so Caitlin walked into my office one day and she said, "There's another study that I'm very interested in." And this is a study that was published in not Lancet, in New England Journal of Medicine, but basically showing that, even though we've developed all these new therapies for ER-positive breast cancer, we still have a big problem. And the big problem is late recurrence. So breast cancer is unusual. I won't say unusual, but it's not typical that when you're defined as cured of your cancer, that 15 and 20 years later, it can actually come back again. And so it turns out that at the Memorial Sloan Kettering, Jose Baselga did a phenomenal thing. He basically sequenced the DNA of every patient that came into Memorial Sloan Kettering, and he and another guy, Sarath Chakrapathi, basically went and looked in aromatase-resistant patients whose tumors are resistant to aromatase inhibitors. Were there any mutations in the estrogen receptor? Now, this was a pretty way out there project because we have sequenced the hell out of DNA in primary tumors and never found an estrogen receptor mutation. But it turns out the difference at Memorial Sloan Kettering is that they were biopsying metastatic lesions. And I'm not going to show you the data, but if I wanted to look at PIK3CA mutations, they're in the primary tumor and they're in the metastatic tumor. So in other words, if I have information about the primary tumor, it informs me later on in life, later on in the treatment regimen, what that patient might respond to. But with respect to ESR1 mutations, they were never found in the primary tumor. Okay? So it turns out now that there's over 40% of patients with aromatase-resistant breast cancer, have mutations in a very, very small area in the ligand-binding domain, the carboxyl terminus of the estrogen receptor. Interestingly enough, the 3 leucines that we talked about earlier are among the 3 leucines that are also mutated quite frequently. And so the issue then is that these mutations reduced the affinity for the only approved ER modulator in this stage, that was fulvestrant, by about 10-fold. Well, I've already told you that we're actually giving women 5 ml injections now in each buttock to try and get a circulating level of that drug that will saturate the receptor. The affinity of these mutations, the affinity for fulvestrant drops by 1 log. There's just no way to deliver enough drug. So fulvestrant doesn't work, and it was shown by John Robertson as well that it doesn't work. So Caitlin walked in and she said, "Donald, we have over 10,000 estrogen receptor modulators in our freezers," because I've been working in this field for a long time, right? And so she wanted permission, rather than working on this failing project, to actually screen, take a shot for the fences. She wanted to screen all of the 10,000 compounds using a robotic system that we have, which took a couple of weeks, okay, to see if there were any that would actually inhibit and be the estrogen receptor the mutated forms of the estrogen receptor. Now, that's a pretty risky project. But all the PIs in the room know that when a student, is desperate, you let them do what they want to do, because you need some time to think about a new project for them, So I let Caitlin off, and I wouldn't be telling you the story if it wasn't a massive success. So let me just describe what these mutations, just on the top left graph, you can see what this looks like. This is what's called a transcription assay. This is a cell where we've put in a cDNA expressing the wild type or the mutant receptors and looked at its transcriptional activity. And this one here will describe to you very quickly what happens in these mutations. The blue line is the transcriptional activity of the wild type estrogen receptor, but the green and the red are the mutants. You can see they no longer need estradiol anymore. They're just constitutively active. Bottom right is the answer. She found a drug in our collection called lasofoxifene. And you can see that lasofoxifene inhibits all of the mutants equally well. The wild type is inhibited, and both of the two most commonly occurring mutations. Unlike fulvestrant, see there's fulvestrant, one log shifted, and like raloxifene, one log shifted. This one was completely insensitive to the mutations. She went and she looked at some of the other mutations, and likewise, whatever way this drug was interacting with the receptor, it didn't care about these mutations. And so this was also interesting for another reason, because I actually discovered this drug in 1992 as a treatment for osteoporosis. That's why it was in my collection. It was developed as a bone-selective estrogen agonist. Again, forget about the terms, okay? So that study there is now in the phase 3 ELAINE trial, okay, which we hope will read out. It's a 500-person phase 3 that we're hoping will read out in the first quarter of 2026. And that's a picture of a graduated Caitlin, whose project number 1 didn't work, but project number 2 got her a PhD. So here's the state of where we are now with SERMs. I update this slide pretty much every month. This up here, you can see that this is a Taxol. This is the very first drug that I described to you that DuPont killed. And then now what we have is a whole series of drugs down here that were all based roughly on the same structural scaffold that Taxol was. I'll tell you a tiny bit in a few slides about camizestrant, which even though elacestrant is the drug that came from my lab is on the market, personally, I think camizestrant is going to come out and be the best one of this class. And I'll tell you why in a second. I think that giredestrant has a 50-50 chance. This is the Roche drug. It failed in a phase 2, but sometimes when things fail, you just put more money, more patients into it, and that's what they did. And imlunestrant, the data read out at San Antonio. I wasn't that impressed. And palazestrant, if it doesn't kill people, it might get approved. This drug, I don't know why this drug has been approved. This drug causes grade 3 and grade 4 neutropenia in patients. I just don't understand how they can actually continue this drug, but that's not my job to make that decision. There's elacestrant is over here because it's slightly different in terms of its mechanistic class because the way it drives the receptor different structure. And I won't go through the rest of them. But the bottom line is that all of these shown in black, okay, were drugs that I gave very high hope for that failed. And it turns out that we now know the reason they failed. It turns out that in 40% of these patients that fail aromatase inhibitors, remember I told you they have these gain-of-function mutations? For some reason, those gain-of-function mutations actually allow all of those drugs in black to actually function as superagonists. So the reason they failed was they probably worked really, really well, okay, in ESR1 wild type, but because our population is so heavily weighted for ESR1 mutation, that's why they probably failed. So I think those drugs could have a second life on wild types, but we don't know that. So that's where the field is right now. And I'm looking, I still have a few minutes, okay? Because what I want to do is I just want to get across one other thing. And I also want to provide you some data to support the argument that I put that that camizestrant might be a particularly good drug. So this slide here summarizes everything that I've got in the slides afterwards so that if I run over, I can tell you, I can summarize it now and tell you what the story is. The story started about a few years ago when I went back to look at the registration trial data for tamoxifen. Remember we started this talk and I tell you about tamoxifen and Craig Jordan. Well, in the registration trial, it showed that the response rate in the first-line setting to tamoxifen was 35%. The overall response rate was 35%. But if they went and they started dividing this up, they found that the response rate in ESR1 wild type patients, ESR1 expressing patients, ER expressing patients, was 28%. No, that's no big surprise. It works in those tumors that have the estrogen receptor. But what was always forgotten is that it worked in 13% of patients who didn't have the estrogen receptor. And when I asked Craig about this, and Craig said, well, that's because we weren't very good at measuring the estrogen receptor. That's a plausible explanation. I said, could I proffer you another explanation? And that is, not all of the activities of these modulators are coming from their cancer cell intrinsic actions, that they're affecting processes extrinsic to the cancer cell that actually can affect the biology of the tumor. He didn't believe me. So what we did was, we actually went and did an experiment. So again, anyone who studies breast tumors in mice, syngeneic models of breast tumors, will know that there are no good models for estrogen receptor positive breast cancer. None. And so my dad used to always say, let the reproducible problem in a system be the first step in the solution. Well, the reproducible problem is, I don't have estrogen receptor, good models for estrogen receptor positive breast cancer. But in some respects, that was good, because what it allowed me to study was the impact of cancer cell extrinsic biology on the biology of a tumor, okay, absent that tumor responding to estrogen receptor. Okay, so here are 4 models. There are 2 models of melanoma, because I'm really interested in sex differences in cancers, in the manifestation of cancers. But here's a EO771 and another one. These are both models loosely of triple negative breast cancer. Okay, and all I want you to see here is that when we knocked out the estrogen receptor in the brain. So remember, we were knocking the receptor out in the brain. The brain, we're trying to influence a peripheral tumor. What you see is that this tumor is stimulated, shown in red, by estradiol. But if you knock out the estrogen receptor, you actually, the tumor grows as if it's been exposed to estradiol. So let me just make sure I drive that point across. We are knocking out the estrogen receptor, actually I'll tell you where, in the arcuate nucleus in the brain. Don't ask me why we did this. It was not for this. It was a purely accidental finding. We knocked it out, and we found that the tumors grew absent estrogen. We went on to look at melanomas, and it's exactly the same. Look at this model here. I mean, this is dramatic. Estrogen has a dramatic effect, but if you knock out the estrogen receptor in this brain, it grows without estrogens. So why is this important for the pharmacology? Think about it. I've just told you that the goal now in estrogen receptor pharmacology with respect to breast cancer is to degrade the estrogen receptor, okay, because of all these signaling pathways. But what happens if I'm degrading the estrogen receptor in the brain? That's the same as genetically knocking out the receptor. And so we said, well, boy, this could be pretty catastrophic, right? Because every company has been trying to develop their SERDs, selective estrogen downregulators, to make them cross the blood-brain barrier. So we did, Suzanne Wardell did this study with Gillian Kayazi, who's another graduate student in my lab. And again, let's just look at this. We hooked up with a company that I was involved with, and they made us a brain-excluded estrogen receptor degrader and one that would penetrate the brain. It doesn't take a rocket scientist to look at. The one that is brain penetrant, okay, stimulates the growth just the same as genetically knocking out the tumor, but the one that is brain excluded works extremely well in expressing the growth of the tumor. This is camizestrant. So camizestrant, which is the AstraZeneca SERD, does not cross the blood-brain barrier. We've measured it. We can't find a molecule of this thing in the brain. And so what I say to you is that even in this one slide, I've told these one little sub-story, I've told you that the estrogen receptor distal to the tumor, okay, is having a direct and dramatic effect on tumor biology. And that now I'm also telling you that that's informative. That piece of that basic finding is informative with respect to how to use these drugs. Obviously, if a female has breast cancer brain metastasis where the tumor is in the brain, then that's the biggest problem, and you deal with that problem, and you allow them to use or encourage the use of drugs that cross the blood-brain barrier. But if you're in the adjuvant setting, or if you're in trying to treat high-risk patients who have no evidence of disease, I would proffer that you don't want a drug that crosses the blood-brain barrier. And so that's my justification right now as to why it works. So before somebody jumps out of their chairs and says, what about elacestrant, the drug that we developed? Remember, this was developed as an estrogen receptor, a brain-specific agonist. By the luck of gods, this thing does cross the blood-brain barrier, but it doesn't antagonize the receptor. It actually activates the receptor. And so this negative feedback loop that we have found that you're destroying by taking away the estrogen receptor, we think we're actually reinforcing by having this ectopic partial agonist activity in LSS strand. So I'm not going to go into the last part of this just because I think I've used enough terms, I've put enough data into the talk to get across a few points. The first point is that I think you'll agree that this was a long project. And I went through a lot of graduate students and postdocs who made their very good careers, chipping away at the understanding how the cell recognizes and responds to estrogens, SERMs, and SERDs. And that was a fundamental platform whereby the screens that I talked to you about were intuitive. They were screens that came because, not just out of the blue, they came and were designed based upon the science that we had uncovered. And so these were very good, I believe, examples of mechanism-based discovery. And that then led to the whole concept of SERDs, which we're happy now has resulted in the approval of one and likely lasofoxifene 2. I think imlunestrant will be approved this year as well, and CAMI probably next year. So it's a really good time for endocrine therapies, given that the last endocrine therapy that was approved before ours was in 1999. So it's been a pretty good advance. And then the last thing with respect to the importance of basic science is that we were, I think, wise enough or lucky enough to realize that even though we had developed these drugs that could really hammer the estrogen receptor within the tumor, there was still more to be done. And the two things that need to be done is understand how the estrogen receptor works in the brain. What's that negative feedback loop? I've got a student, Gillian, working on that. And again, I was telling someone this morning, I don't expect that paper to see the light of day for a couple of years because there's a lot of work to be done there. And then finally, I'm not going to show the data, but I'll tell you that one of the most exciting projects we've worked on, and we've just published on recently, one paper published and one coming out in JCI, that estrogen receptor in immune cells is extremely important. And that we have knocked out the estrogen receptor in every single immune cell. And the only one where we don't see any biology is the T cell. We see indirect effects on T cells through effects on dendritic cells and NK cells and such like, but no effect on T cells. And so the next time I'm invited, if I am, I will tell you about the ER immunology story, whereby we've now actually shown that we can manipulate the immune system, and that now has allowed us to think about, and we're doing, clinical trials combining endocrine therapies with immune checkpoint inhibitors outside of breast cancer. And so now we're starting to wander into this area of gender-based differences in response to immune checkpoint inhibitors. So I hope I've also convinced you that I have one talent, and that's recruiting really, really good students and postdocs. Thank you very much for your attention. Sorry, just take my wares.
Absolutely phenomenal.
Thank you.
I'll probably take one question. We're going to have a panel later on for more questions. Maybe one. Yeah. James?
Fantastic talk.
Thank you.
I'm just curious on what your opinion is that, based on your data, that we really should be doing a lot more screening for mutations prior to treatment to try and define the correct biomarkers for the right patients.
Yeah. So, obviously, I could stand up in the bully pulpit and talk about the benefits of precision medicine, which is basically what you're talking about. In the case of the estrogen receptor, it's going to be a little bit difficult, right? Because we are only finding the mutations, I'm talking about anyway, in metastatic disease. So unless you're asking me, do you think, and I hope you're asking me this because I think it's a great idea, should we biopsy patients with metastatic disease and then look at the differences in the mutation profiles in metastatic disease versus primary? I think all of the data would definitely support that. The problem is getting metastatic biopsies into the standard of care. That's the problem. And in the States, and I don't know how it is in Ireland, is getting that reimbursed has been the issue. It's been limiting us. But it's not rocket science. If the mutations only occur in the lesion, you need to be able to find those mutations, and the only way to do that is to screen the metastatic. Now there's a company in Boston called Precede Biosciences, and I consult for them, so that's my conflict. But they have developed a phenomenal liquid biopsy. So there's lots of companies out there. They'll basically take blood, they'll find ctDNA, and they say, there's tumor DNA. Problem is that's way too late. Okay, when you have at least enough tumor DNA to measure, it's way too late. But what they're doing is that they've developed an assay that is so sensitive that they can probably detect the tumor DNA when there's only a few metastatic cells, very few. So that's one thing. But the second thing is they don't just look at the presence of the DNA. They actually it's a really unique technology. They sequence all the active enhancers in that DNA. And they've developed a technology that's so sensitive that they can look at the active. So not only can they say, yes, Mrs. Jones has ctDNA, but they can say Mrs. Jones' ctDNA enhancer profile says she needs to have a MYC inhibitor. She needs to have a PI3 kinase inhibitor. So that, to me, is where this is going. And then one last thing. Jean Bidard, God, I'm pregnant of his name, his first name, Jean-Pierre Bidard. He did a fantastic study called the PADO-1 trial. I love it. So the PADO-1 trial was a case of where, if you're on an endocrine therapy for breast cancer, what would happen is you'd go onto an aromatase inhibitor, you get your 6-month checkups, and you say, sorry, Mrs. Jones, your tumor has progressed. We're going to put you onto fulvestrant, okay? And then we're going to put you onto CDK4/6 inhibitor or whatever. What the PADO-1 did was they actually went and, rather than using radiographic progression, they actually went and profiled for ESR1 mutations in circulating tumor DNA prior to any biopsy. Okay, and what they did was that and prior to radiographic progression, they switched the patients then onto fulvestrant. The difference in the PFS is massive. So, yeah, you've got me. We're both hopefully in the same crowd. I love this idea, and I think that if we could just find ways to incorporate it into standard of care and in practice, that we'd make great strides. Okay, thank you. Sorry, I promise you I'm going here.
So our third talk in this session is from Lorraine Walsh. Lorraine is a consultant radiation oncologist at the Midwestern Cancer Centre in UHL here in Limerick. She's got a keen interest in MRI-guided brachytherapy for cervical cancers. I'm sure she's going to tell us all about brachytherapy and how it's the microneedles with radiation, isn't it? Is that a good description or not? No? Okay. MRI-guided brachytherapy for cervical cancers, re-irradiation of gynecological malignancies, and the adoption of novel radiotherapy technologies such as intensity-modulated and image-guided radiotherapy to reduce normal tissue toxicity during breast, lung, and pelvic irradiation. So, phenomenal to have you. Thank you, Lorraine. Thank you.
Thanks very much, and thanks for asking me to speak. I don't think I'm going to do anything as good as what the speakers before me have said, but anyway, my talk is going to be brief. I work as a radiation oncologist in the Midwestern Radiation Oncology Centre based in University Hospital Limerick. I'm going to give a brief overview as to what that service entails initially, and I think this is important to touch on some of the challenges in commencing research from our department, and then going to talk about 3 specific areas of research that we're currently working on. So the Midwestern Radiation Oncology Centre in University Hospital Limerick is an unusual department. It opened in 2005, and it's a joint venture between the Mater Private Hospital, University Hospital Limerick, run by the HSE, and then JP McManus's Midwestern Hospitals Development Trust. So the reason why the centre opened was because there was no public radiation oncology facility in Limerick at the time, and patients had to travel to Cork or Dublin for their radiotherapy, which for a lot of women who were receiving breast cancer radiotherapy, it meant that they had to leave their families, and sometimes they opted for more extensive surgery so that they could avoid radiotherapy because it caused such upset in the family to have them move out of home. So JP McManus and the Mater Private came together and developed the Midwestern Radiation Oncology Centre, which is based on the grounds of University Hospital Limerick in the Midwestern Cancer Centre. It is a very busy department. We have treated about 16,000 patients since it opened, so we're treating nearly 1,000 patients a year. There's 2 radiation oncology consultants, myself and my colleague Dr. Al-Bassouni. The treatment unit, we have 2 treatment machines, and they work 12 to 14 hours per day, which is probably 50% longer than most treatment units would work. Nonetheless, it's a really nice department. We're fully staffed, we've got good staff retention rates, and we're very technologically advanced. We are fully equipped with all forms of treatment, intensity modulated radiation, brachytherapy, MRI-guided treatment facilities. So it is a very well-developed department and technologically advanced, but very busy. We replaced one of our machines in 2023 to the Varian Halcyon platform, and this is the most up-to-date and fastest treatment unit in the country. So it is able to treat patients in what sometimes would be 45 minutes. We can now treat patients in less than 10 minutes using the Halcyon platform. Now, it's not a treatment unit that's suitable for all tumour types. It does have some limitations, but it has given us an extra 50% capacity on that machine. So, it allows us to keep working through our patient load. And currently, we are replacing our other machine, and we are going to be replacing that. It was currently being replaced, and it will be open in the summer with a Varian TrueBeam. So the reason why I mentioned all of that is I suppose it has been difficult to start research in our small little department in Limerick, which is kind of owned by 3 separate entities. So what is radiotherapy, first of all? Well, radiotherapy is the field devoted to the treatment of benign and malignant diseases in adults and children with ionising radiation. We don't typically treat children in our department. We do treat some benign conditions, but obviously it's mainly malignant diseases. So in contrast to diagnostic radiology, where they're using low energy X-rays to image a patient, radiation oncology uses X-rays as well, but they're much higher energy. So they're this part of the spectrum up here. Does this work? No, this one does, yeah. So here's our electromagnetic spectrum. This is visible light here, UV light. These are the X-rays that are used in the diagnostic radiology department, and more energetic than that are the gamma rays that we use to treat cancers. So these are these are some of our radiation therapists. This is our most up-to-date treatment machine, the Halcyon. It differs a little bit from the traditional linear accelerator in that it has a kind of a donut-shaped bore, and the patient lies here, and they cannot see the actual gantry moving around. And this is going to be our new machine that's opening in the summertime. So once the radiation, I suppose, irradiates the patient, it causes excitation and ionisation of the DNA, which causes double-strand breaks in the tumour cells, and the tumour cells die. Obviously, that can also harm the normal tissues and lead to side effects. But normal tissues are genetically different, as we all know, and they have the ability to repair and repopulate and reoxygenate and recover from the harmful effects of radiation. So I suppose this is a little bit negative, but there have been challenges to starting research in our department. And our challenges they're not just local to us. There has been challenges in, I suppose, developing radiation oncology research throughout the country. And I suppose part of that is because there are only 5 public radiotherapy facilities in Ireland, and there are a number of private facilities, and we all kind of, I suppose, worked a little bit in silos and a little bit disjointed, and all very, very busy, a lack of consultants, a ton of patients, and it has been hard to, with the best intentions, to develop research and meaningful research. Insurance has been an issue as well, I suppose, in terms of, particularly in our department, because we're employed and run by the Mater Private, but on the grounds of UHL, and then we have the Midwestern Hospital Development Trust kind of there as well. It has been difficult to get proper clarity as to who's insuring the patient and us as well as the researchers. And then obviously time, access to the treatment machines, like research on 2 very busy linear accelerators who are already operating more than 50% more than what they should be doing. It can be difficult to justify doing research on the machines if you have an ever-mounting patient list. So COVID was a good time because during COVID it was recognised nationally that radiation oncology research in Ireland had to improve. And Professor John Armstrong, who's a very eminent radiation oncologist in Ireland, he suggested that we develop the group called IROG, which is the Irish Research Radiation Oncology Group. So this meant that the 5 public radiotherapy centres came together and some of the private centres as well, so UPMC in Cork and Waterford and ourselves in Limerick, to do trials nationally, so that me applying for ethics approval in Limerick no longer had to be done. This was all done nationally and in a much more fast and efficient way, and everyone was working together with a standardised approach, and it has worked very, very well. So they were very successful in obtaining HRB funding. They worked very closely with Cancer Trials Ireland. I think the feeling during COVID was that Cancer Trials Ireland was doing a great job of the medical oncology clinical trials, but that radiation oncology, I suppose, wasn't getting the same importance attached to it. So that's where IROG came into being, and it's working really well, really well. It's only in its infancy, IROG, so I suppose it made us in Limerick feel a little bit better about ourselves, that we weren't that wasn't just us that were, not doing the research properly. It was good to see that other centres and much bigger centres with much greater critical mass of staff were also running into the same problems in conducting meaningful research. So IROG has successfully now brought together a critical mass of people with expertise in clinical research, trial coordination, data methodology. So we have physicists, doctors, radiation therapists, nurses and allied health professionals, all of whom are needed to conduct proper radiation oncology research. And the good thing about IROG is it not only allows us to do national trials, but it also has allowed us to conduct trials with international groups as well, and I'm going to mention a couple of those now. IROG is looking at not only new technologies, so obviously radiation oncology is very technologically influenced subject, so looking at different ways in which you can treat patients, different energies, different treatment machines, different doses per fraction, but it's also looking at the combination of drugs and radiotherapy. It's looking at omitting radiotherapy in certain breast cancer patients who have a very favourable prognosis, and it's also looking at educating and training the research team nationally. So this year was a very good year for us in the Midwestern Radiation Oncology Department, as we finally were approved and recruited some patients onto this very important international radiation oncology trial in prostate cancer. So the PACE Nodes trial is a UK-based trial, but it's been conducted internationally. It's a phase 3 randomised trial of stereotactic radiotherapy to the prostate gland alone versus stereotactic radiotherapy to the prostate gland and the pelvic lymph nodes in patients who have high-risk localised prostate cancer. So traditionally, and probably in most departments all over the world, the treatment for prostate cancer when using radiotherapy is at a minimum 4 weeks of pelvic radiation. That has changed in recent years. It used to be 7.5 weeks of radiotherapy, so 37 fractions or 39 fractions. It has now reduced down to 20 fractions, so that takes 4 weeks to deliver. It's Monday to Friday for 4 weeks. And that's probably the most commonly used fractionation schema when patients are receiving prostate radiotherapy. Stereotactic radiotherapy is a little bit different. what you are doing with stereotactic radiotherapy is you're giving a much, much higher dose per fraction to the tumour. So it allows you to complete the treatment in a much shorter period of time. And it is proven to work, and lots of patients are receiving stereotactic radiotherapy for certain forms of prostate cancer. Not all departments are capable of doing stereotactic radiotherapy. It requires a huge workload on all of the multidisciplinary team because you're giving out a high dose per fraction. You have to be absolutely certain that you're treating the correct part of the patient each time you turn on the treatment machine. So it requires a lot of work in advance and a lot of time on the table to ensure that what you're delivering is the correct and safe dose of radiation. So this trial, the PACE-NOSE trial, is looking at something a little bit more novel. It's looking at either just giving the stereotactic radiotherapy to the prostate gland or giving stereotactic radiotherapy to the prostate gland plus the pelvic lymph nodes. So this trial is ongoing at the moment internationally, and we've recruited a couple of patients to it here in Limerick, which is great. It's been led by this guy, Sunil Jain. He's based up in Belfast. So he's a radiation oncologist, and his main area of interest is prostate cancer. During COVID, when patients didn't want to come for any form of treatment to hospitals, this publication was fast-tracked. This is a UK-based trial which looked at hypofractionated breast radiotherapy. And what hypofractionation means is a higher dose per fraction, meaning that you deliver the overall total dose in a shorter period of time. So with the hypofractionated fast-forward breast trial, the researchers found that 1 week of breast radiotherapy was as good as 3 weeks of breast radiotherapy in terms of breast cancer outcome, and it was associated with no increased risk in side effects. So all of the radiotherapy departments all around the world, because it was in the time during COVID, we rushed and implemented this form of treatment. And we still do it very, very commonly, where we no longer give 3 weeks of radiotherapy to certain breast cancer patients. We've been able to hypofractionate it down to 1 week of treatment. So this study is a new study following on from that called the Fast Forward Breast Boost study, and that is going to be looking at can we continue to deliver the treatment over 1 week, but also at the same time give what's called a simultaneous integrated boost to the tumour cavity. So this study is opening in Ireland at the moment, and we have applied to be included, and we're hoping to hear about that in a couple of weeks. And it should be a very straightforward study to now embark on because we have the support of IROG. And that is the schema for that study. And finally, I know Emma Jude Lyons and Leonard O'Sullivan presented last year at this very meeting about Emma Jude's research on 3D printed bespoke breast prosthesis for patients post-mastectomy. So we have now had her first case report published. Well, it will be published in June. This is a case report of Emma Jude's study looking at 3D printing bespoke breast prosthesis in patients who are post-mastectomy. And this follows on from a time when this very lady, she was a patient of mine, and she came back to me in the breast clinic after she'd had her treatment, and she flung her silicone breast prosthesis onto the bed when I was examining her, and it was a ton weight. And I said, I think we can do better than that. So I approached Leonard's team, and we worked with Emma Jude, and there's a huge team working on it at the moment and developing the most, patient compliant breast prosthesis for patients. So it's a work in progress, but it's very exciting because we're seeing that patients are really very, very happy with the fit and the weight and the texture of these prostheses as compared to the generic spongy or silicone heavy prosthesis that they would normally be prescribed. That's it.
Lorraine, thanks so much. Again, I'm taking questions at the end when we have the panel, but like your work, and thank you so much for coming here, because I know like the folks that are in physics, they're doing the Raman, I know Sinead that's doing the sensors for radiation. This is really like an active area of research within the university, so it's great. Thank you for coming. And our last speaker within this session, I'm delighted to have PhD student Kelsey Galty, who's in Amira Mahdi's lab presenting. And Kelsey is a first-year PhD student studying breast cancer. Her research is funded by HRI at the UL School of Medicine, and she's supervised by Amira and also by Kieran McGorty. The aim of her research is to build three-dimensional co-culture models to better represent the tumour microenvironment, particularly the role of adipocytes in the cancer therapy response. Kelsey aims to use co-culture models and single-cell RNA sequencing to study and devise strategies to overcome endocrine therapy resistance in estrogen receptor positive breast cancer. So coming full cycle around to breast cancer. So hopefully this will be a phenomenal talk just to sort of bring us full cycle on this session in solid tumours. So thank you, Kelsey.
Thank you for that introduction, and very good job to the previous presenters. I would like to thank Donald and Alwyn for kind of giving a background. You'll all be experts on the background of my project then. So I'll get started. As we know now, a majority of breast cancer cases are estrogen receptor positive, 70% of the breast cancer cases. And the traditional hormonal therapies consist of aromatase inhibitors and selective estrogen modulators. What we're seeing in patients with cancer that returns is they build resistance to these traditional therapies. As such, there are selective estrogen receptor degraders like fulvestrant that was developed in 2001. However, those offer poor bioavailability and are administered with intramuscular injections, which are painful for the patients, as previously mentioned. So next generation SERDs, which are orally available in pill form, are being developed, and there's around 13 at the moment, but there's only one that is FDA approved, which is the elacestrant for the estrogen receptor mutation linked with resistance. So we have a problem here of many of these being in development, but only one of them is approved. And this is because preclinical trials are very isolated and simple and do not focus on aspects like the resistance or the tumor microenvironment. And the preclinical results will be promising, but this doesn't translate to the patient trials and clinical trials. So the objective of my project is to combine the tumor microenvironment and the resistance piece into making better models for these developing hormonal therapies. So a major component of breast tissue are adipose tissue, and the tumor and adipose tissue coexist together, and these interactions have proven to fuel the cancer behavior. So the goals of my research are to build an accurate model including the resistance and also the 3D aspect of tumors that we see in patients, to include the interactions between cancer and adipose cells, and to identify pharmaceutical targets and combination from those interactions. So in my first work package, we do a 3D co-culture spheroid of cancer cell lines and adipocytes, along with conferring resistance and seeing what changes in these models. This will be analyzed using single-cell RNA sequencing to determine gene expression differences in the treatment-sensitive and the treatment-resistant models, and then deriving drug candidate matching with the SERDs and combination therapies, And then this will be tested in combination drug matrix assays with the developing SERDs and the targeted therapy. So an update on a bit of my time in the lab has been on optimizing the 3D spheroids, and we've been using microtissue molds and agarose, which has been easy to replicate, inexpensive, and easy to reuse. And the agarose surface repels the cells and causes them to compact with each other to form spheroids, which we've done with MCF7 cells alone. And we've been monitoring the growth to determine if the small or the large microtissue molds are better to use, and we find that the small ones are more consistent, the large ones are more variable. And days 4 through 7, the growth is most stabilized, which is good for the histology processing of these microtissue molds. And then we can stain them and do histology slides, for example, with DAPI and TRIT-C, and already starting to see some interesting patterns here. And finally, we hope to add in the adipose cells to this process as well. Thank you. And if you would like to connect with me on LinkedIn, I left a QR code there. If you have any questions or suggestions, my email's there as well. Thank you very much.
Can I ask all of the speakers to come down to the chairs here for the panel? And while they're doing that, let's give everybody a clap, because that was a phenomenal session. I think, have we got a mic that we're going to, for oh, okay. Is this one for the audience, or is this one for okay, great. Wow, that was brilliant. Oh, wow. Okay, so I'm probably going to kick off. Is this on? Is this on? It's on now. Okay. Whoa, that was really, really cool. I loved that session. Thank you, everybody, so, so much. I have my questions here, so I'm just going to get it up. Sorry. Wow. So I suppose one question I had was actually, Donald's, in your work, and thinking of kind of gender and cancer, and then bringing into kind of maybe, you know, how you're building models and everything like this, because we know adipocytes and we know these models are really important. And obviously, like with gender, we're thinking of, you know, the hormones, but also that, you know, we see most cancers in postmenopausal women. Where we know that the hormones in like pre and post menopause are very different. Like, do we have models that recapitulate pre and post menopause?
Well, I think the question is, you know, you can develop models of pre and post, but whether they actually recapitulate the disease is the issue. The answer to your question is no. I mean, you know, when you look at something like osteoporosis, for instance, yes, we have. We can develop models where estrogen deprivation will give the same changes in bone morphology in a rat models, for instance, a human. But I think, as you were indicating, you know, is that there are no good models really for estrogen receptor positive breast cancer outside of cell models. There's nothing being more predictive of the actions of estrogen receptor modulators than a plain old MCF7 cell in a dish. But as I hope I got across, is that just represents the cancer cell intrinsic, and we really need to develop models where we can look at more holistic impacts on the tumor growth coming from the brain. The bone actually feeds to the tumor as well. The immune system feeds the tumor, and we're not reading on those. But if you don't mind, because we'll hand it over to Alvin, we talked about this morning. there's a really great opportunity, I think, right now to actually look at not only differences in the incidence, but also in the progression and the morbidity of diseases as a function of gender. Okay, it is clear that there are some cancers that are clearly sex biased. Okay, the outcome, and not just because of the biology. Breast cancer obviously is more biased in females than in males, but there's a physical reason for that. But if you look at melanoma, for instance, the outcomes are much, much different. Women, premenopausal women, do much worse than postmenopausal women, and premenopausal women do much worse than any male. Males always do better. And this is normalized for anything like exposure to carcinogens and things like that. There are fundamental differences in the response. The bad news is, or the good news, whichever, all of that research will have to be done in Ireland now, because I can't do that in the States anymore.
I know that it's banned now, isn't it? Any diversity research.
Yeah, I've actually gone and scoured my grants for anything interesting and taken it out. but I think it is an incredibly important issue. And I think that just with respect to your talk today, we have a study going on right now looking at endocrine therapy as an adjuvant to to radiation therapy in lung cancer, because it turns out that the process that by which a lot of lung cancer tumors repair themselves, called a process called efferocytosis, can be actually blocked by estrogen receptor modulators. So let's think beyond the traditional cancers and other ways that we can take this observation of nature and turn it into therapies. I know you have some comments on that probably as well.
I will. We spoke about this earlier. I didn't get to talk about it, but another stream of our work is looking at the gender dimension in bladder cancer. And again, taking away that, carcinogens factor, because it tends to be 4 times more likely in men. People tend to attribute this to the fact that men smoke a lot more. But when you actually normalize that and take away the carcinogens, you still have 4 times higher incidence in men. And also women have worse progression and a much later diagnosis of bladder cancer. So being able to treat biological males and females differently is really something that we have to understand. And again, even breaking that down into pre and postmenopausal, because bladder cancer diagnosis at quite a late stage, you're going to have women that are premenopausal and postmenopausal. So being able to understand the hormone dynamics there is really important. That's something that we are starting to work on at the moment, to try and establish whether there is anything targetable, anything different in males versus females in terms of behaviour and progression of the tumour cells.
Yeah. I mean, there must be differences. I mean, if you think back to lung cancer and like the patients who have certain EGFR mutations and they're non-smoking females of Asian extraction, I mean, that's been proven. And there's targeted drugs now, and these women can present, at a young age with stage 4 lung cancer, and they can live for years with targeted drugs, whereas previously they would have been, written off to palliative care, and that was it. And that's a prime example of how mutations have been found in these certain cancers, and targeted drugs have been developed to, and these patients do very well.
Kelsey, just thinking about your adipose, we know that like post-menopause, and I'm going to open this up to everybody else, this is still on the gender. Adipose tissue is a major source of estradiol post-menopause, and it's the major source of estrogen. How do you think your work with the adipose will maybe inform that?
I think so far I haven't done a ton of work with the single cell RNA sequencing, but already with that you can get a lot of information from different databases, kind of with the pre and post menopausal adipose tissues that have been sequenced, and using the differences in that, I think, will be a big highlight to studying those differences.
Do something with cell exchange there. Okay, I'm going to open this up to everybody else. I'm not going to dominate the conversation.
You might have to.
I have about 10 questions here, so I'm happy to dominate if we need to.
It's just a brief question, really. I suppose we've talked a lot about targeted treatments and hormonal treatments, but as regards the tumour microenvironment and maybe adiposity, is there a different approach when it comes to radiotherapy, Lorraine, depending on the patient's makeup? Is there or any mutations, or have you any different approach, sort of personalised or targeted?
Not really. No, although there can be logistical issues. But I even had a patient yesterday who's just too big to fit on the treatment machine. So we do have those issues. But there isn't anything, no, not really, not that I'm aware of, that would, affect the patient's sensitivity to radiotherapy depending on how adipose or not they were.
Or even prior medication or, their chemotherapy or targeted therapy?
No, not that I'm aware of, but maybe it's something.
Yeah, but there should, I think, there's one of the things that we're looking at actually in radiation is whether the difference between concomitant introduction of endocrine therapies with radiation, or because classically what we do is we do radiate and then we follow. And actually we just did a study where we're starting to deconvolute the data right now. We're actually, you could interpret it in saying there's a really good idea to doing concomitant. So those sort of things I think will change the field. Yeah.
Yeah, and people who came to the meeting we had in January on data, there was a phenomenal talk there from Paul Nagy on how they're integrating the radiation images into the real world data studies across like millions of patients and hospitals. So yes, so I think there's going to be some really cool real world data on radiation as well. Amira.
Hi, well done everyone on your talks. They're really interesting and informative. So I have a question for Donald about the orally available SERDs. So I think this is something I tried to figure out, well, I tried to look up, but where do you think these orally available SERDs will sit in the current treatment landscape for breast cancer? And there's a lot of options there. Didn't epilepsy just got approved in the UK? Like there's
who got approved?
The PI3 kinase inhibitor.
Oh, yeah.
So
epilepsy.
How do we fit them in?
So you raise, I was actually on a phone call with an analyst the day before I left because he asked me exactly the same question because they're trying to figure out what all of these SERMs and SERDs, what all these targeted therapy comes. It's a quagmire in terms of how these diseases are going to be treated. I think, in reference to your question earlier, I think some of this is going to come from from precision medicine. Okay, what is the physical makeup of the tumor or the metastatic lesion in that particular patient? I think what we've come from is an area where we do endocrine therapy. That fails. We do endocrine therapy plus a CDK4/6 inhibitor. Now we move on to yet another endocrine therapy. We may move on to a PIK3CA mutation inhibitor like alpelisib, and then we basically go to classical chemotherapy. That's the way it is right now. I think what's going to happen is that we're going to get to the point that some of these agents are going to be brought forward, into the first line setting. Okay, there's already, a lot of patients are treated now with CDK4/6 inhibitors in the first line setting. If that's a good idea or a bad idea, I'm not really sure because you're taking away an option that patients do respond very well to downstream. So I don't think that the metastatic setting is going to change much more in the sense that we're going to find mutations, we're going to treat those mutations less. But where the big change is going to be is in the frontline settings. I still say that endocrine therapy will be the cornerstone of interventions for a very long time. The question is, which endocrine therapy? I think it's going to be really very difficult to beat an aromatase inhibitor. I think that, it's the Parsifal trial where they actually looked at fulvestrant versus an aromatase inhibitor plus a CDK4/6 inhibitor in the front line. You'd need a green laser pointer to separate the lines. Aromatase inhibitors are dollars per month, whereas the new SERDs are not dollars per month. So that's an economic question, even though there might be a slight difference in efficacy. Then just not to go too far, but I think I mentioned the PADA-1 trial, these early interventional studies. I think that's going to be the way we're going to go. We're going to be looking to when do we change patients from drug X to drug Y, rather than letting progression-free survival determined by radiographic measurements be the endpoint. I think that's going to be. And then I really, really believe, and I wasn't joking, I've said it in pretty much every lecture I've ever given in the last, given in the last couple of years, is that I really think that we need to stop this, to change the mindset from I want to treat your cancer to I want to cure your cancer. Nothing of what I talked about today is going to cure a cancer. It's just going to treat the cancer and treat it as a chronic disease. And so I think the only way that we can do that is to harness the immune system. I just don't see any other way of that. So.
So I'm just going to open up one question that I think I've had these discussions with Dave Livingston. I've had these discussions with every breast cancer researcher that and it's one that to me I've never got a good answer for. So, I'm just going to throw it out there for the sake of it. Which is when we think of germline mutations and BRCA1, and the model for so many years has been that, it's BRCA1 in the epithelial cell that's driving the cancer. How we know, but we know that defect in DNA repair that is causing that cancer in that epithelial cell, that DNA defect repair is actually in every other cell as well. It's in all the immune cells, it's in the stroma, it's in the adipose, it's in every single cell. So the question is what's the contribution of those other cells to the cancer? And I think, Lorraine, when you're thinking about sort of radiation oncology and inducing DNA strand breaks, is there something that you see, or when you're thinking about how we treat this and how we actually model this, what do we do with germline mutations and in the cancer? And this is
I mean, it's funny that Dave, of course, passed away a couple of years ago, but he was a wonderful man. But the question is actually, it's a very interesting question because, as you said, the BRCA1 and BRCA2 are germline mutations. They're now not only just important in breast, but also in prostate cancer as well. The bottom line, the question, though, has never been answered since Andy Futrell discovered, the Mary Clay King discovered this in 1991. Nobody understands why is it that every single cell has the exact same mutation, but only breast, ovarian, and prostate actually manifest as disease? Huh? Well, pancreas is, there is a rare cases of pancreas as well. But the bottom line is that nobody really knows. The original idea was, they're the rapidly proliferating cells. That doesn't seem to be the case. And so I think it could be the microenvironment that these cells are in is also contributing to it. Bottom line, though, is that the other thing which is interesting is that we treat with PARP inhibitors, and they're relatively benign. So it's the same thing, is that we're still even inhibiting the PARP. BRCA sensitizes you to PARP inhibitors, and yet I'm always surprised that they're not more toxic as well. So I think again, I'm telling us all to get our mind out of the cancer cell intrinsic biology and think of it as a part of an ecosystem, and we need to understand the ecosystem.
Anybody else want to go into
make a comment?
Mutations?
Make a comment about that one. You're right.
I think that there's also there is studies there showing that BRCA1 could be important in immunology, in development of immune cells. Yeah, and so the question I think I always have there is how much of it is that the immune environment might also be compromised and then not able to manage the damage so well, and that's the reason why we see cancer earlier in these women.
I think that once you mention the word immune system in any of these discussions, you're on the right track.
I think so too. Actually, one of the things we did not so long ago is we were looking at the gene tree family for all of the PD1, CTLA4. What you have there within that tree is all of the butyrophilins. Yeah. And the butyrophilin genes, butyrophilin is butter. Okay, and that's because they occur in the first breast milk that occurs. It's like that buttery yellow colour breast milk, that colostrum that occurs. And these all evolved way back as far as platypus, and so they're really old genes, and these were important in immune from very, very, very early, but they were originally delivered via breast milk. So is there connections there that we don't even know about? Just throwing it out there. Any last question, or I think we'll just wrap up? I know people are dying for tea. So which way are we going? Okay. So with that, I think I'll, I always like to end on a question rather than an answer. So I want to thank everybody for coming to this first session. We'll have a quick break, and we're going to come back to what is going to be an incredible session on blood cancers, which will be led, and immunology, and lots more immunology, because we know immunology is the cornerstone of everything. Yes, and Liz is nodding here. So thank you so much, and we'll see you back in, what time? 15 minutes? Okay, thank you.
Okay, so just following on, thank you, Ruth, for the great introduction. So I'm an immunologist, and I am interested in immunology of different patient cohorts, looking at understanding immunology in people. Okay, so in CLL, we have, I'm going to use this, we have the proliferation of B cells and what happens here is these proliferating B cells, first of all, take up space, so you have fewer of the other immune cells developing, but they also secrete a whole pile of immunomodulatory molecules that can influence the function and development of many different types of cells in your immune system. So innate and adaptive immunity is impacted. Okay, so this leaves patients susceptible to infection, and they also respond very poorly to many different vaccines. So we are working on this, and we're not alone working on this. There's large groups of interested researchers throughout the world working on this, and we're very lucky to be collaborating with a group called ERIC, which is the European Initiative on CLL. And we've recently put together a project called CLL to Cure. And these are this is our network. So there's a good representation from Scandinavia, Germany, and the Netherlands. And together between all of our groups, we have expertise in many different areas of biology, and an expertise on T cells and metabolism. And what we're bringing to this group is an expertise on innate immunity and investigation of the microbiome in these patients. And this, as Brian, who's doing a PhD with us, will tell you, is a really understudied area. We don't really understand innate immunity, so pathogen recognition and pathogen detection in these patients and how disease affects it. And interestingly, the drugs that really target B cell proliferation in these patients also target key enzymes such as BTK. They're involved in key pathways like the toll-like receptor signaling pathways that are involved in pathogen recognition. So while these drugs are excellent, they have further impact on patient immunity. So if we're going to cure these patients, we need to really understand the immune systems. So we're going to do this in a number of different ways. So first of all, we know these CLL cells have key driver mutations. And depending on the mutations the cells have, this impacts the proteins and the genes that they express. So different clones of CLL cells will have different microenvironments. And we really don't understand the link between the mutation and the microenvironment. So the one way we're approaching this is in collaboration with a group called Phantom. So Phantom is a long-running collaboration to understand the human genome, apparently, more or less. So it's in its 6th iteration, and it's led by Piero Carnici, who works between the Riken Institute in Japan and the Human Technopole in Milan. So this group in Phantom 6 are studying and mapping chromatin and in 3D. So looking at chromatin protein interactions, chromatin RNA interactions, using 2 techniques called Hi-C and radical-seq, which are 2 very specific ways of investigating 3D structure and long non-coding RNA. So this is an important link between mutation and gene expression and protein production in the tumor microenvironment. So we have been very lucky to be able to submit a number of our patient samples to this study, and we've selected them based on different driver mutations, and this will allow us to investigate the impact of key driver mutations on the tumor microenvironment. And this is in collaboration with Dr. Pauline Robe, who is an expert on CLL genetics, and our postdoc here, Louisa, who is working with Deirdre and a number of the clinical trials team to recruit the patients, isolate and purify the CLL cells, and fix them in such a way that we can process them for Hi-C and radical-seq. So Louisa is also very involved in our collaboration with the University of Ulster that has been facilitated by the All-Ireland Cancer Research Programme at Start. So this is a north-south research programme, and we're working with two researchers in particular in the two PIs in the University of Ulster, Kyle and Andrew, who Kyle's an expert in blood cancer and Andrew is a microbiome expert. And we have two postdocs on this project, Louisa and Anya. And together they will map the immune profile in patients and match this with the microbiome. So again, microbiome in your gut and in your skin and in your body dictates how your immune system operates. It's well studied in a number of solid tumours, particularly colorectal cancer, but there's really no information at all. Very, very few limited studies on, what the microbiome is in blood cancer patients. And CLL patients in particular tend to be older, and we know as we age, our microbiome becomes less and less diverse. So it's very likely that this narrow microbiome is a contributing factor to how their immune systems are developing. But it's a very under-researched and explored topic, and it's something that we're beginning to try and establish. So they are just a couple of our key projects that we are investigating with national and international collaborators. And again, thanks to the Biobank and the Clinical Trials Unit, we have access to really valuable patient material. And without this, we wouldn't be able to make progress in diagnostics and, translating new therapies to the patients. So thank you very much. So to follow this, I'm really delighted to introduce a PhD student who's been working with me for the last year, Brian Leeson. And Brian is focusing in particular on neutrophils, which are a really important innate immune cell. And we want to see how the role these cells play in infection risk in patients with CLL.
Hi. Thank you for the introduction and for the comprehensive overview of the cancer and also of immune suppression, which actually happens as a consequence. So as we have actually seen, the patients with CLL are immune compromised, which happens both because of their cancer and also because of the treatments. And this really puts them at an increased risk of opportunistic infections, which is a huge challenge for patients and has a negative impact on their quality of life. So I'm interested in neutrophils, which haven't really been studied, I suppose, as much as any other immune cells have, but they're critical first responders in infection. So they can ingest pathogens by phagocytosis. They produce these really toxic antimicrobial proteins by degranulation, and they also produce neutrophil extracellular traps, which actually help to trap, and obviously they kill pathogens, which helps to actually prevent the spread of infection. So my first aim was to kind of see, to actually see if neutrophils can respond to infection in patients with CLL. So I reviewed the literature to actually see, have there been any neutrophil functional or phenotyping studies. So I searched PubMed and Scopus and Web of science, and I imported all of these results into Covenants, which is a systematic review software. And after my title and abstract screening and then full text review, I ended up with 43 studies which were included in the review. So overall, neutrophils appear to be dysfunctional in that so in the bone marrow, the counts are actually higher before treatment, but then after treatment, with, just as an example, BTK inhibitors, both the mature and immature neutrophil counts are extremely lowered, which is obviously, this is a challenge for the patients. And also then in the peripheral blood, neutrophils have an enhanced capacity to actually produce these nets, so those extra cellular traps, which these can both enhance CLL, cell survival, while also increasing T cell suppression. And then at the site of infection, then neutrophils have impaired capacity to both migrate and also to actually kill pathogens. And all of this is also severely inhibited after treatments. So these studies aren't perfect, and I did observe that there are some gaps in the current knowledge. So these studies, they had quite small patient cohorts, and also they only explored one neutrophil, say, function in each study, as opposed to, say, like the overall kind of immune phenotyping, which is preferable. And also there was significant variation in experimental design in each study, so it's hard to extrapolate between them. So this brings me to my next aim, which was to determine, okay, how can we systematically screen neutrophil responses to infection? And in order to determine this, we had to see basically what are the markers of neutrophil activation in response to, say, bacterial or viral infections. So in order to see what these markers are, I had to actually search for some publicly available bulk RNA sequencing data sets. All of these used purified peripheral blood neutrophils. You'll observe that approximately half of them are from infections and half of them are from cancers, but there aren't any actually from either patients with CLL or from any other blood cancer, because they don't exist apparently. So I then took these data sets, which were all on Geo, and I then ran them through an established transcriptomics pipeline using the tools that are on the slide. I used R for my differential expression, and then I used the tools that you can see here for my functional enrichment. So in patients with COVID, we observed an overall enrichment for genes that actually mediated neutrophil degranulation. In particular, genes like CD177, OLFM4, and others, which haven't previously been studied in patients with CLL. So then when you compare the neutrophils in patients with bacterial infections, you can see it's a completely altered signature. Now, this is potentially because we only had 2 data sets, and obviously pneumonia and sepsis are quite different, but all the same, we observed that there are 2 genes that were actually in common between the 2, CD244 and GSTM4. They didn't appear to have any direct interactions. You can see they have been studied in other conditions, but haven't been studied in neutrophils in CLL. So they are potentially novel. And then when we compare in solid cancers, which is obviously an approximation here, but we can see that there is also that increase in neutrophil degranulation, in particular MMP9 and CD177, and also that a few of these genes were also specifically in the neutrophil tertiary granules, which is just a specific type of proteins. So then I put all the data sets together, and I observed that there were 22 significant genes that actually came up, well, we'll say in the majority of the data sets. I believe it was 5 out of the 9. You can see that once again we have this in enrichment for neutrophil degranulation, and in particular I want to highlight CD177, which has actually been associated with a distinct subpopulation of neutrophils, which actually have enhanced they're highly activated, and they have enhanced capacity for migration and pathogen killing. So in conclusion, my review highlighted that there is most probably some type of neutrophil dysfunction in patients with CLL, but we aren't sure if it's true, we'll say, for like more broadly because of the limitations of the previous studies. So I then highlighted some potential novel markers. Now, these are all at the RNA level, so we'll have to actually validate these in publicly available proteomics data sets. And we're also hoping to then do some in vitro validation of, we'll say, the key markers, and hopefully then we'll progress to patients. And I just want to thank my supervisors and my colleague, Dr. Louisa Silva, and my FYP students who had their presentation last week. And so I think that's it. Thank you.
Well done, Brian. Thank you very much. And we can take a question or two at the end on that. So we'll move on now, a bit of change of pace, back over to the clinical world. We have Fidelma Hackett, our advanced nurse practitioner who works in the haematology department in UHL, presenting on CLL from the patient, carer and practitioner perspective with Professor Pauline Meskel, myself and Professor Rose Galvin supervising.
And thank you for your time today. so we'll get started. So a little change really, chronic lymphocytic leukemia, which we've spoken about already, but from the patient, the care, and from the practitioner perspective. So more on the survivorship end of the clinical research that is ongoing at UHL at the moment. So just a bit of an overview there. So chronic lymphocytic leukemia, we know that it's a slow-growing malignancy. So it is a blood cancer. Very varied clinical presentation, often an incidental finding, which brings its own issues. The person is often asymptomatic and very well. So we've heard about the unique treatment landscape now with all of these types of cancers. But in particular, this one can be difficult for patients because the decision to treat really depends on the clinical stage, the prognostic factors and how the patient is. So there's no advantage to early stage disease. So we've got a growing number of patients now that get a diagnosis of cancer, but the treatment plan is clinical observation only or watch and wait until there's evidence of progressive disease. So this brings its own issues, as you can imagine. So we've heard of huge advances today in treatment and prognostication, and in the hematology world, and particularly in CLL, over the last 10 years, we've had huge advances, particularly in the targeted therapies and the oral anti-cancer medications, which the patients, we've many patients on. Now, this has led to reduced CLL mortality and increasing survival, which is a good news story, obviously. But why do this research then? Well, there's many studies in cancer survivorship, over the last decade, but the focus mainly is on solid tumours and living beyond cancer. So once you've been cured of your cancer, living beyond that. So CLL is treatable, but yet it remains incurable, not for long after listening today to all the lovely presentations. But there's periods of remission and relapse. So the focus of the treatment is not cure, but improving the quality of life and, these patients living longer. So the concept of survivorship is problematic for this group because they don't live beyond their cancer. They live with this cancer. So living beyond the diagnosis really is an increasingly important survivorship issue. It brings challenges. They're living with CLL now as a chronic cancer, and that's not something that maybe our services are set up for. The long-term disease surveillance, there's treatment decision-making, with these periods of remission and relapse. And then there's an increased patient and care responsibility, particularly with these targeted therapies. They're prescribed in the hospital, they're managed in the hospital, they're dispensed in the community, but they're administered by the patient at home with the support of the caregiver, usually the spouse at home is giving out the tablets and bringing people to appointments, etc. Then the CLL morbidity, which Ruth spoke about, is becoming increasingly an issue because people are living longer. So the infection risk is a huge issue for us, and now the secondary cancer risk. So these patients are living for many years. Many of them are still on these targeted therapies 10 years later, but now they're developing prostate cancer, they're developing skin cancer. So that has an issue as well. The healthcare challenges then. The big thing really is managing this chronic disease course. There's a growing patient population of survivors. There's also a growing number of diagnoses, I would say. We've 1 or 2 new patients each week in clinic. How are we going to manage this group of patients who are on watch and wait and active surveillance? Our services are not designed, they're not set up for that. We need to balance the priority for acute cases, patients that need to see the consultant, and acute relapses, and patients with acute leukemia maybe. So we need to balance our resources and our support services to both support and help these patients, but also to balance our priorities. And for the clinician then, taking into account treatment sequencing, we've heard that today, that's hugely important now. The treatment costs are significant, and of course this is an older aged population, so the treatment toxicity is an issue. So what my research really, my main aim is to understand what is the experience like for these patients with CLL, what's it like for the caregivers at home, and what's it like now for the practitioners in Ireland, looking after this disease, with the current treatment landscape. So my focus will be on living with, coping with, and managing CLL. And really, the focus or the reason for that is to optimise what support strategies do these people with CLL need. So how can we improve their quality of life and the outcomes? And ultimately, I suppose, to co-design what's a more suitable care pathway, and a model of care for these patients. So patient engagement and participation in research is really vital. We are really committed to that in UHL Cancer Services, and we have a lovely PPI group. So I've also established a PPI panel for the lifetime of the PhD. Hopefully it won't be 6 years. My plan is to do it a little bit sooner. And they have been fantastic. So 3 patients have a diagnosis of CLL, and I have 2 caregivers who have spouses with CLL. There's also CLL Ireland Patient Advocacy Group lead and board members, and they're also involved in the research. So I presented the initial research plan to them, and that led to the addition of caregivers being involved, which we hadn't thought of at the beginning. It was going to be patients and practitioners. And they're, at the moment, they are looking at my data from my current qualitative review, and I'm meeting with them on Friday for them to give their opinion on so it's a collaborative data analysis we're doing with that, which is fantastic. I'm sure they'll come up with plenty of things for me. So my first piece of work was conducting a systematic qualitative review, and I was looking at primary studies, qualitative studies on the experiences of people with CLL, their care partners and practitioners. So it was primary qualitative studies, was my review question. After an extensive research, I had over 1,700 papers. But finally, after title and abstract screening and full text screening, I have 20 publications. So 11 studies from 17 publications are with patients, just 1 study with clinicians, a Greek study, and 2 studies with caregivers. So just the findings, and as I say, I'm still in the analysis stage, but the sub-themes or the early data that's coming through, navigating diagnosis, that is a serious, that is an issue for patients. The impact of diagnosis, obviously with any cancer, shock, anxiety, but this one's a little bit different because there's trying to understand and accept this diagnosis without treatment. There's a lot of disbelief. It's counterintuitive. Get cancer early, get treatment early. The diagnosis disclosure is coming through as a big issue. And patients really we know from clinical practice, they're not bringing this up in conversation. So they're kind of managing this on their own, if you like, keeping the diagnosis private, strategizing it. Who do I tell? When do I tell? Do I tell people at work? These types of issues. So they really need support here. And then this chronic nature of the illness, they feel that is misjudged. And often people refer to it as good cancer, which it's not always good, for people. So the support network and coping mechanisms are coming through. There's certainly support from the healthcare team, and they need that, they want that, they're looking for that. But there's also the importance of the support system outside of the hospital. So the spouse, the friends, the family, the community, peer support, the support groups, and online support. And then the different types of coping strategies that people are using, as well as counselling, philosophical, religious, that's a big issue. And then physical exercise, things like that. So the impact then, what is the impact of this disease on a person living with CLL? So their physical function, certainly, role function, and fatigue is coming out really strongly. Now, we don't really have much support services for fatigue, I would say, and it is an issue with a lot of different cancers. So that maybe is something that we look at. I was at a conference recently in the UK, and they had a fatigue management clinic for their MPN patients. So the patients were sent off to a clinic. Now, that would be amazing. There's nothing like that in Ireland. So maybe you'll hear that in my next talk here. The psychological impact of living with CLL, anxiety and emotional toll, but uncertainty is the big thing because these patients don't know when they're going to need treatment. So they're trying to plan for the future, and that uncertainty certainly has an effect. COVID-19 pandemic really intensified all of these feelings because the infection risk, as we've heard, is a big issue. So patients have, always had to socially isolate, I suppose, and always had social limitations, but that really increased their feelings of vulnerability. So the findings then from the 2 studies with caregivers. So they're also trying to navigate this CLL management and understanding the treatment options. They're heavily involved in helping patients manage these treatments at home. The responsibility for, giving the chemotherapy in the hospital has now changed to responsibility of the person at home. So they need support. They're looking for support from peers and from healthcare professionals, and they have a feeling of this constant caring, which is often an issue with a chronic illness or a chronic disease. Their social connections and social isolation is an issue. They are also socially restricted because they're trying to support the person at home. And then this perception and awareness of CLL is coming through as an issue. It's seen as an invisible disease. The person looks quite well, so they don't always get that support from friends and family and that understanding. And then just one study on practitioners. And this was a Greek study, and it was specifically looking at exploring communication in clinical practice, so one specific area. So disclosure of medical information is an issue, the medical decision-making and emotional support. So I thought of particular interest there was maintaining support for relationships with patients was important, but it was particularly given limited access to mental health professionals. So, that might be something that we might find here also. And then medical decision making was balancing the guidelines, with individualised care for these patients and what might suit them. So a whole person kind of care model. So in summary, I suppose patients and caregivers emphasise this is a complex disease for them. The social isolation, the information support networks, ways of coping, these are all the issues that are coming through. So it's important that we look at ways of giving these patients information and supporting them throughout the phases of the CLL, particularly given the growing numbers. But the additional, I suppose, significance of navigation through diagnosis is something that we do need to look at, because often these patients are discharged maybe back to the GP, or maybe they just come once a year. So that's a long time to be trying to navigate a diagnosis before you meet a healthcare professional again who's a specialist in the area. So that is something that is of interest really in asking practitioners around the country, what are they doing to manage these patients? So the haematologist, the focus was on patient-centred care and effective communication and support. But certainly looking at the studies, it's understudied from both the caregiver aspect and also understudied from the practitioner. So that is the plan really for the research now going forward. So I'm looking at addressing these perspectives with qualitative studies with patients, with caregivers in the Midwest, but also looking to the healthcare professionals around the country through the CLL Alliance. So of particular interest is these support considerations and what coping strategies, and the patients that cope very well, what can we do with the patients maybe that have unmet needs here, they're living alone maybe, they don't have a support network, how can we support these patients? And then relapse disease is certainly an area that's not been studied very well. Now patients are on their second and third and fourth line treatment because there are such good treatments, but how can we support and cope? How can we help them cope? And ultimately, I suppose, to develop an end-to-end clinical care pathway that's not a survivorship model of care for a cancer that's cured, but that's also not a chronic disease management module. They don't quite fit into either, but they're somewhere in the middle, and maybe pieces from both of those. So we'll be looking to talk to the patients about that, what's acceptable to them and what's feasible then in different settings. So that will be year 3. I think that's it. So thank you very much for your time today, and thank you to my supervisor, Professor Ruth Clifford, and also to my supervisor, Professor Pauline Meskel, who's here in the audience somewhere as well. Thank you.
Thank you very much, Fidelma. So our next speaker is Aileen McMahon, and Aileen is a senior medical scientist in the Haematology Diagnostic Laboratory in UHL, and she's undertaking a part-time PhD supervised by Ruth Clifford and myself. And Aileen will tell you about improving diagnostics for lymphoma patients.
Thanks, Liz. So the title of our research is Feasibility of ctDNA and Immune Response Analyses in a Population of Irish Patients with High-Risk Diffuse Large B-Cell Lymphoma. So just a quick background, diffuse large B-cell lymphoma, or DLBCL, is the most common non-Hodgkin lymphoma, accounts for around 30%. At diagnosis, it's classified as germinal center B-cell, activated B-cell type, or unclassified, and this is using immunohistochemistry. It is curable with the standard of care treatment, which is given to all patients, ORTOP, it's immunochemotherapy, but around 30 to 40% of patients will be refractory to treatment or will relapse. We do know that the ABC subtype is associated with a poorer prognosis and a higher degree of immune dysregulation, but even with this information, it can be difficult to predict how individual patients will respond to treatment. So there is an unmet clinical need in the early identification of patients with refractory disease. And if we can identify these patients earlier, then there's potential to change them to an alternative treatment and reduce unnecessary treatment-related toxicity, which is more important, or significantly important for the older or frail patients. So in recent years, with increased genetic testing in research labs, there are now 7 molecular subtypes of DLBCL which have been identified. So this paves the way for a more targeted approach to treatment. And some mutations such as mediation CD79B have been identified as associated with resistance to RTOP. However, this genetic information is generally not available to Irish clinicians because minimal genetic testing is done on the tissue biopsy at diagnosis. So currently, how do we assess how a patient is responding to treatment? So an interim PET or CT scan is done, and then an end-of-treatment PET scan is done to assess how well the patient has responded. But these scans can be difficult to interpret due to immune cell infiltration. So sometimes this means then patients need additional testing like repeat scans or biopsies in order for the clinician to be able to formulate their care plan. So how can we improve early detection of refractory disease? So a liquid biopsy is a simple peripheral blood sample, which is a non-invasive alternative to tissue biopsy. Using the liquid biopsy, we can detect and analyse the circulation tumour DNA, which is DNA released from tumour cells into the peripheral blood as they die. And because it's a non-invasive sample, it allows for serial sampling because it's not so easy to go back and re-biopsy or retake a tissue biopsy. So with circulating tumour DNA, if you can identify the mutations, then we can assign a molecular classification. You can quantify and track the circulating tumour DNA in response to treatment and monitor for emerging resistance mutations. And then we know that the tumour microenvironment is increasingly becoming a focus of research in DLBCL. So using peripheral blood samples, there will be circulating immune markers there which should reflect what's happening in the tumour microenvironment. So can we quantify and track inflammatory markers or immune cell types in response to treatment as well? So to find out more, we conducted a scoping review to see what knowledge was already out there on how ctDNA and immune markers can be used to assess treatment response and predict outcomes in these patients undergoing first-line treatment. I won't go through all of this in detail, but just briefly, the main findings were that there is a real lack of standardization in the laboratory methods used for ctDNA analysis. There's a lot of custom panels used and different sequencing platforms. So it definitely would need to be standardized to be introduced for clinical practice. A number of studies noted that the circulating tumour DNA results do improve interpretation of the interim PET scan. And there was one study which reported that undetectable circulating tumour DNA at end of treatment reduced the need for patients to go through additional testing. And the most significant time points in terms of prognostic value to take samples are at pre-treatment, cycle 2 day 1, cycle 3 day 1, and end of treatment. And then in terms of the immune markers, again, there was a wide variety of immune markers which are analysed, but some did come up more commonly than others. And then the main kind of finding was that the increased cytokines at diagnosis is associated with inferior survival. Then a couple of studies found that increasing monocytic myeloid-derived suppressor cells was associated with superior survival, but most of the studies on immune markers only took samples at baseline, so there's a need there to look at taking samples as patients progress through treatment. And then one study combined the inflammatory markers and circulating tumour DNA and found an inflamed serum protein profile in combination with a high ctDNA burden at diagnosis was associated with a poor prognosis. So for our study then, the hypothesis is that in patients with DLBCL, tracking circulating immune markers in combination with ctDNA can aid in the earlier identification of patients at high risk for refractory disease on first-line treatment. So our aims are to explore the association between ctDNA and the circulating immune markers in response to treatment in these patients to determine if this relationship can be used to aid in the early identification of high-risk patients, and then to investigate the feasibility of introducing circulating tumour DNA analysis for patients with DLBCL attending Irish cancer centres. So our plan is to recruit 20 to 30 patients. We'll take serial peripheral blood samples at pre-treatment, cycle 3, day 1, and end of treatment, and perform circulating tumour DNA and perform next-generation sequencing for circulating tumour DNA analysis on these samples, and then perform flow cytometry and ELISA for the immune markers. And then we will also, in parallel, take the diagnostic tissue biopsy and perform next-generation sequencing on this and immunohistochemistry to study the tumour microenvironment. So that will provide a comparison for the baseline peripheral blood samples. And we plan to recruit patients over 18 to 24 months and follow them up for at least 6 months post-treatment, hopefully up to 18 months, depending on when they're recruited. And then we will look for correlation between the circulating tumour DNA results and the immune markers at the different time points, then see if there's a correlation with these results at the different time points with patient outcomes. And then also look to see if there are any specific mutations in the ctDNA that are associated with increased inflammatory markers. And so the next step is that we are now open for recruitment, finally, and that's it.
Fantastic, Aileen, that's great. And then we'll move on to our last talk. Clivert, who has travelled all the way down from Queen's. Thank you very much for making the effort to come in person. He's going to present on his PhD in the blood cancer work package of the eHealth Hub, led out by Aideen and Mark Lawler, on connecting all island clinical and molecular data for lymphoid blood cancers to illuminate immune dysfunction and drug resistance. And Clivert supervised by Professor Ian Overton at Queen's, myself here, and Siobhan Glavey, haematologist at Beaumont RCSI. Thank you, Clivert.
Hi, good afternoon. So my name is Clivert Boachidua, and a student at Queen's University, and my PhD research is connecting all island clinical and molecular data for lymphoid blood cancers to eliminate immune dysfunction and drug resistance. So I'm to link and integrate CLL and multiple myeloma data from Northern Ireland and the Republic of Ireland, and after that use it to understand the immune dysfunction and drug resistance in those two cancers. So one of the key problems with health data is data standardization. Data standardization is the process of transforming data to a consistent and uniform format. Hospitals worldwide use different systems to collect healthcare data, and these different systems create dissimilar data sets. And it makes it difficult for researchers to come together to analyze data. Sharing healthcare data is also important as it improves collaborative research. So our approach is to transform these dissimilar data sets to a common format called the OMOP Common Data Model. OMOP stands for Observational Medical Outcomes Partnership. And after transforming these dissimilar data sets to this common format, we will then analyze and share the results. We are using the OMOP Common Data Model because it has been accepted as the gold standard for transforming health data by the European Medicines Agency. And the OMOP Common Data Model was developed by a community in the US called the ODC community. So the reason why health data standardization is important is that it increases collaborations among researchers, it simplifies the sharing of healthcare data, it supports interoperability, and overall it improves patients' outcome because we have more data to analyze. So before we transformed our data to the OMOP Common Data Model, we did a landscape and gap analysis to understand the current state of all island data. So we selected clinicians and data managers, and we sent them a questionnaire to ask them about the tools that they use to collect their cancer data, data sources, data quality, and conditions inhibiting data sharing. We also reviewed key landscaping papers like the e-health strategy for Ireland, cancer data moving forward by Dr. Michael Quinn, and we did some informal interviews in Beaumont Hospital, University Hospital Limerick, and the Blood Cancer Biobank Ireland. So for the landscape analysis in Ireland, we found out that hospitals use different systems to collect cancer patients' data. Paper notes are still used to collect patients' data. The individual health identifier is not fully used, but the successful implementation of the e-health strategy for Ireland will improve data collection and sharing. In Northern Ireland, hospitals use the same system to collect cancer patients' information, and the full rollout of the Encompass program, which uses EPIC as electronic healthcare record, will improve data sharing and collection. So we have also developed this computational workflow to map our data to the OMOP common data model. So in the data extraction phase, we will extract data from electronic health record and clinical notes to a database, and we use Whitewrite, which is a tool developed by the ODC community to screen the data for descriptive statistics. We also will do quality control and semantic enrichment, and we are using the R package called the EHD prep. We will also do data transformation using some tools by the ODC community, that is Athena, Usagi, Rabbit in a Hat, and we will use the data build to write SQL code for the data standardization. We also do quality control after transforming the data using Atlas, and data quality dashboard. Then for the final stage, we will do data federation and analysis. So we have access to the Blood Cancer Biobank Ireland data, and that is what we have mapped to the OMOP common data model. So this is the heat map of the Blood Cancer Biobank Ireland data, and it has missing values, and which is shown in the yellow phase of the heat map. And we have also been able to map the data to the OMOP common data model, and we are using a tool by the ODC community to view the mapped data set. And in the BCBI data, we can see the number of persons in the BCBI data. We can use this Atlas tool to calculate for the incidence rate. Also, we can use it to define cohorts for clinical trials. And to assess how we've performed when we transform the data to the OMOP common data model, we use the data quality dashboard also by the ODC community to check how well we performed. So for plausibility, we got 95%. For conformance to the OMOP common data model standard, we got 100%. And for completeness, we got 95%. So obviously, there are rooms for improvement. So for my future work, I'm in the last stage of getting data from CLL, from University Hospital Limerick, and I'll map it to the OMOP common data model. And I've improved the EHD prep R package, and I'm waiting to test it on other data sets. And I will demonstrate a federated access to clinical data across hospital sites. And after that, I will analyze the data for immune dysfunction in CLL and resistance mechanisms in multiple myeloma. So I want to acknowledge my supervisors and my funding body. Thanks very much.
We might just do a very quick question and answer session up the front, just to give you the honour. So if you want to prepare a few questions yourselves, if there's any from the audience, myself and Liz can get started. Okay, pressing questions from the audience. Okay, James.
As always, I love your work from everyone, actually. But I'm specifically interested in yours, Ruth, when you were talking about the growth of the blood tumours and the factors that they secrete. Is there any evidence that the factors secreted actually have adverse effects on any of the antibiotics used when you're talking about they succumb to secondary infections. Is it just a response to the tumour cells, or are they actually modulating the effect of the antibiotics?
So I think they're affecting the cells around them, aren't they? Not necessarily the antibiotics. That's not studied, to be honest. So we don't know that. But something our patients suffer from is hypogammaglobulinemia, which is a low level of immunoglobulins. And that is something our patients have chronically, even at an early stage of their CLL, and that can lead to an immunocompromised state for some patients. So certainly, CLL cells inhibiting the microenvironment, but also other B cells, particularly plasma cells that make immunoglobulins. There isn't any evidence that, I understand the question about affecting antibiotics directly, but there's no evidence for that, or no, I mean, we haven't really looked into that, but potentially, I suppose. I don't know if you want to comment.
Turns out it is on. So the CLL B cells secrete a huge amount of interleukin-10. So interleukin-10 will skew your myeloid cells towards myeloid-derived suppressor cell lineages, and they also amplify T regulatory responses. So they're switching on all the things that downregulate your immune system. So that is, if you can block that interleukin-10 by the production, that will help your other immune cells deal with infection. So they're modulating all the immune cells in the microenvironment, really.
I might just, is that okay, James? Sorry. Okay. Don't.
Aileen, I was very interested in your study. I think actually it's going to be a gold mine, I think, to analyse it afterwards. If you're just looking at just the ctDNA, the measurements, so maybe it wasn't quite clear to me. So I understand why you want to measure the ctDNA. I mean, it's a very good barometer of relapse. But what do you want to do with that information? Is it descriptive and qualitative right now, and then go from there? Or what's the endpoint that you want to correlate with the ctDNA?
So, with the ctDNA, there's a group, an American group, David Kurtz, who described a molecular response that you should see and that correlates with the better prognosis. So from baseline, when the ctDNA is measured, a 2 log decrease after the first cycle, or a 2.5 log decrease after the second cycle, it means that the patient is doing well and has a better prognosis. So that's basically, if we're going to measure the cycle 3 day 1, so we want to see a 2.5 log decrease in the ctDNA.
Gotcha.
So if we wanted to pick out the patients who don't have that decrease, and then that means that they are not responding to treatment as well. So in the future, in probably other studies, then maybe it could be looked at, like a different treatment could be looked at for those patients.
Yeah, so I suppose it's just it's thought to be a more sensitive biomarker than radiology, which is what we currently use, and that you could come in at an earlier time point and highlight patients who aren't responding at the correct pace and change tack, and then you could also set it up as a biomarker study with a new intervention, maybe a BITE or bispecific treatment in second line for elderly patients who will get chemotherapy toxicity, etc. Yeah. So it's very much looking at the response, and then the immune angle then is the novel angle. Yeah.
So one of the things I'm hoping that maybe we can talk offline, but there's a lot more information in ctDNA than just the measurement of the actual DNA. You can actually tell a lot about the disease course and things like that. And in breast cancer, what they're doing now is, of course, they're not just looking at enumerating ctDNA. And in fact, drop in ctDNA is obviously in a barometer of response there. But people like Dan Hayes and that at Ohio State, they've actually been looking at different aspects, qualitative differences in the ctDNA to actually predict responses and potential outcomes to new drug interventions. So.
Yeah, and there are certain mutations which have been identified, which wouldn't routinely be tested for, I suppose, in patients. So, if we could do that using the ctDNA, then obviously that would be.
But I mean, the nice thing about your study is that you're going to have a biobank afterwards that you can, as science moves forward, you can iteratively go back to that.
Yeah.
Yeah, it's great.
I have a question for Brian. It was a wonderful presentation, Brian. Great work. My question is about the neutrophil extracellular traps, and I have very minimal knowledge about neutrophils because I work on platelets and cancer. Yeah. So I was under the notion that these nets are like substances released out by the neutrophils, and they localize somewhere in the tumor microenvironment, and they promote the platelet aggregation and migration and metastasis. But is there any specific anatomical focus for them in CLL? Because there might not be a solid tumor microenvironment there. So.
Yeah, so we've actually had a conversation as to if we think it's happening, say, out in the periphery, or if they're possibly infiltration, say, into the lymph nodes. All the studies that I had in the review, I think all of them, apart from one, they took peripheral blood neutrophils and they activated them, and they then measured the capacity to actually produce the nets. I believe there was only one study that used immunohistochemistry to detect. So they actually detected MPO, which is the myeloperoxidase, and they also looked at histones, citrulinated histones C3, which is also an indication of the production of the nets, and that was detected in 37 patients with CLL, and I believe also about 20 odd patients with DL BCL, I believe, but it was only in one study. There hasn't been anything else, so it's absolutely we are interested, and we're also actually hoping to possibly detect them in existing blood smears actually. So we actually have an instrument in the hospital that we believe we could be able to detect them in. So there's there's archived smears in hematology that we're hoping we could be able to perhaps.
And we just did. So we had a biomedical student in School of Medicine did their final year project on this with Aileen's supervision and mine. So we have an automated instrument looking at blood film morphology that we can detect it. And in the past, we actually thought that these were, you get smudge cells that are friable lymphocytes on the peripheral blood smear, but actually we were counting all of these as smudge cells, lymphocytes, but more than 50% of them are actually neutrophils that look like these historical basket cells, and they've been teased apart by flow cytometry in certain research studies, but there are certain parameters that we've been able to pick up in the lab, either through direct morphology or the DI60 digital instrument. So Brian will continue that work, but that's certainly something. But looking at it in lymph nodes is trickier, isn't it? But that's something that we can potentially do if we have the lymph nodes. Generally speaking, for CLL patients, we just have peripheral blood samples, and we don't necessarily need lymph node biopsies, so they're limited.
Thank you.
Aideen.
I think the folks at BD Biosciences here on campus might well be interested in working with this, because we know that 10x genomics single cell platforms actually normally lose neutrophils, so that you don't actually capture the neutrophils properly on 10x. Whereas I think this is one of the areas where actually the BD single cell platforms actually do, and I think they have data on that to show that they capture the neutrophils, whereas the 10x don't.
BD.
Yeah. So I think they will probably be quite excited.
Yeah. And they've been working with a group in Germany who were part of, the My Uniter cost action. So they were working with a group I know in Germany, and they have some really nice preliminary data on neutrophils. There's very few studies with neutrophils because a lot of immune studies are done on biobanked peripheral blood mononuclear cells, of which the neutrophils are not part of that prep. So a difficulty, but an advantage. We need fresh samples for our studies. So that's why there were very few studies out there. But there is they're very important. They're 70% of your immune cells. They're the first cells to the site of infection, and they really coordinate a whole lot of activity. So yeah, we need to know.
Thanks, everyone. Fantastic work. My question actually is for Fidelma, again around the patient perspective. And you mentioned, Fidelma, the issues around the watch and wait and challenges around anxiety and that. In the literature, is there a better way to do the watch in terms of like outcomes for patients and less anxiety in that?
Patients probably need more help than we're giving them managing this area. And we know from the literature that difficulty with the watch and wait can go on for many years. And patients are just coming to the hospital maybe once a year. So there should be an intervention, something to help them, support them through that, particularly for that first year. Over time, of course, this helps. And maybe there's a way of assessing patients psychologically each clinic visit, and then picking out those patients maybe who are more at risk and referring them on to our psycho-oncology colleagues. So there's definitely something in that, really. And I'd say starting with the assessment piece and maybe the informational support piece as well. Yeah.
Thank you very much. So I just might end with a question for Clivert, unless there's any other pressing questions in the audience. So based on your work to date, because you've been looking at the different infrastructures in the North and in the Republic, are there any key glaring differences or commonalities that are helping us with our data collection from your gap analysis?
So in the South here, although different hospitals use different systems, there are like common tools like T-Pro for clinical typing, and also I think the NCI also will be rolled out very soon. So there are common tools that are being used which could help. Whereas like in Northern Ireland, like all the hospitals use the same system, and the introduction of the electronic health record will also help with data collection and sharing. Yeah.
Thank you very much. So unless there's any other questions, do you want to close, Aideen? Yep. All right. Thank you, everybody, for that great session.
Thanks, everyone. I'm just giving a couple of few updates on kind of, this is really short. It will literally take 2 minutes, but I just wanted to highlight some really good work and some good news stories. So the first thing is congratulations to PIs who were awarded higher education research equipment grants. The LDCRC chose to co-fund some applications to increase the opportunities for funding. And there's been 3 projects funded. So the MADI lab, congratulations, Amira. Is Amira still here? Okay, congratulations, Kelsey. There's a new drug screening platform that will be purchased by the MADI lab, the Synergy HTX, which was an automated drug screening platform. Congratulations to is Owen still here? Owen Canan? Okay, we've lost a couple of people. It's late, I know. And it's sunny. Congratulations to Owen's lab. They've got, they will actually be able to purchase a new 3D bioprinter. So this will allow to bioprint cells on matrices to create in vitro systems. Alwyn can help answer questions on that if you want. And I put in for a grant for computing for the LDCRC PIs. So we are now working with the ITD to actually purchase some computers, and we'll create accounts. So there'll actually be, for the first time, some data infrastructure for hopefully sharing data and for analysis across LDCRC PIs. At the moment, that will just be CPU computing. We're not going to make the GPU stuff available to everybody, but we are going to enhance the capacity within my lab, at least for GPU compute, which is really important for the digital pathology and the imaging data, as Ahmed will quite happily tell you as he's hammering the GPUs we have at the moment. Doing a lot of digital pathology. And onto the digital pathology side of it, we've been mostly using images that the Ryan lab, Conor Ryan's lab and my lab have been doing quite a lot recently on analysis and machine learning methods on digital pathology images. The work in our lab, I can tell you, is looking at trying to look at perineural invasions specifically and seeing if there's associations between perineural invasions and then markers of T-cell response, such as the amount and the locations of infiltrating T-cells. And towards that, we're absolutely thrilled that Maura Laval, who's a breast pathologist at UHL, was successful in her application for Spark Award funding. She's got almost 100 grand to buy a new scanner. And she will work to, and I think the scanner system that they're going with is the Vendetta DP200. And their plan is to produce digital images of all breast pathology images and then scale out to other solid tumours. So we will now have a biobank of digital images of tumours, which we can hopefully then apply our machine learning methods on. So this is going to be a really, really nice merger of both UL and UHL capacities. And therefore, it's going to be really important to align with the biobanks, which are, thankfully, thanks to Liz and to James and to Alana and to everybody else who's been really fundamental and pivotal, that's really bad words there, in developing the biobank. So thank you, everybody who's got the biobank up and running as well. Congratulations to Amira, another Amira award. So she's doing absolutely amazing, who got a CanServe platform award. So CanServe is a European funded project that provides your EU researchers access to facilities that they don't have in their home institution. And so she will have access to patient-derived co-culture models in Graz in Austria and drug screening services in Valencia in Spain through this CanServe platform. And this is a great opportunity for people, and I'm sure we'll have Amira present a little bit time of how she's used the CanServe platform to expand her research and collaborations. Speaking of research and collaborations, I'm kind of excited and kind of like nervous at what it is, but I am now the head of node for Elixir Ireland. So that means Ireland's development of Elixir has actually been quite minimal to date, but in other EU countries, Elixir is the organisation that actually coordinates life science data infrastructure. So that's compute, platforms, training, tools, databases, and such like for life sciences, health and life sciences. The government is committed in this funding cycle, and it was again announced during the week in the, DFARS have their strategy that's open for comment at the moment, but they've committed in that strategy to fund research data infrastructure. So there will be funding for this upcoming, and we're hoping that through kind of the bioinformatics network, we have quite a big existing bioinformatics networks that spans all universities, many of the TUs and Chagas, that together we can put in a coordinated response there to make sure that Ireland has the bioinformatics infrastructure to advance life science and health. Towards another grant towards this, we are members of the 1+ million genomes Genome of Ireland project, and this will be 1,200 individuals representative throughout Ireland will be sequenced as part of the 1+MG programme, and we're minor players in this. This has really been led out by our CSI and Trinity, but their goal is that this will be a representative healthy population throughout Ireland, but there will be subsequent parts of the 1+MG which will be focused on rare disease and cancer and different pathologies where we know the genome and genetics is really important. And finally, I'll finish where I started, which is an advert and a plug. Next Thursday, the lab showcase. Sign up your lab. It's in the afternoon. It will be basically talking about what in vitro cancer model systems we have. Not just like what's in the set, what we're doing as far as in the lab, but also digital pathology capacity for, and like it will range from, you know, how can we scan a cell? So things like the folks that in physics that are developing Raman imaging, and the folks in electrical engineering that are developing fiber optic light sensors, so we could do real-time cell imaging, through to basically what are those 2D cell banks that we should have in UL. How can we develop better 3D models? And I think Alwyn spoke really nicely about that, and so did Kelsey today. Through to how could we develop patient avatars, through to how can we work with SSPC and NIBRT to develop functional precision medicine platforms. And functional precision medicine is this idea of drug screening on patient tumours, really pioneered by Tony Latai's group at Dana-Farber. And hopefully we'll get our phenomenal brains together, and we'll at this one grand challenge day or hackathon that we're going to have, and we'll see, can we develop ideas collectively together to reduce the cost of organoids. Amira has written a really nice proposal on the problem that maybe we can do that. Can we develop patient avatars or organ-on-a-chip systems? Alwyn really developed some really nice ideas there today. And can we actually work with our collaborators, think of how we might put in a Cancer Grand Challenge grant? This is really well worth doing. It is 25 to 30 million in funding. These are really, really big, massively prestigious grants. Let's see. Let's aim high. Let's see where we can go. You know, this is what we should be doing. Even if we don't get there, if we get shortlisted, we'll get 30 grand to actually develop the proposal. So this is, if we're not in, we can't win. Let's aim high, let's go for it, let's put our heads together and see how we can develop the best application possible. Okay, thank you. And I have just remembered I have a beautiful plants that are sitting for both you, and I've forgotten to bring them. So my thank you to the amazing and the formidable Trisha English here, who is just absolutely amazing for organising everything. She keeps me organised, and that is no mean task. And then thank you to Katie Kremen.
I don't, sorry.
It's probably true. I'm sorry. I'm too difficult to challenge. And thanks also to Katie, who helped so much with everything as well, and really supported Trisha in this. So thank you to Katie and Trisha. And thanks to our phenomenal keynote and all the speakers. Give yourselves a massive round of applause.